Hello, everybody. I'm looking a little different today because during the move to the new apartment, somebody, meaning me, damaged my mic stand. So instead of mounting my phone on my phone stand, I've mounted my mic. So I'm reactive PNG-fi today. Hello, everybody. Thank you for joining me. Hello, Eric. Hello, Seed. Hello, Archie and Steven. Hello, Hero and Nicholas. Welcome, Clockwork and Surprise Pants. Robert, hello. Mapping, welcome. Charis, welcome. Hello. I saw that you just collabed with Z. How did that go? From what I heard from Z, I heard you all had a whole lot of fun. All right. So what are we talking here about today? Oh, those were words. You can tell that I'm also freshly awoken. Today, we are going to be talking about this paper that Clockwork actually recommended in the Discord. Exclamation point Discord if you aren't in there already, about the mitochondrial origins of the pressure to sleep. And to me, this was a very clickbait. This is the kind of clickbait title. And I'm gonna be honest, I had a very negative reaction at first. This struck me as kind of sensationalist. And I think it's an interesting opportunity to show you whether or not I have my mind changed from a place of skepticism, right? And because what I want to model for people is not necessarily this is how this is what the science is. I'm trying to model how to responsibly and in good faith engage with the science, right? So even though I think this paper looks like clickbait, I am open actually to either changing my own definition of what I believe the origins of the pressure to sleep should have proof of, like the level of proof that I would need. I'm willing to change my mind depending on the arguments that this paper is making. So I'll say that I'm skeptical, but not outright cynical, right? I think that's a nice place to be. And actually reading the paper will, I think, either I'll dig my heels in deeper and get annoyed, which is something. I kind of don't want that to happen, believe it or not. Or I'll have my mind changed, and I think that's also quite nice, I think. Yeah. When you read the discussion too, the whole thing smelled more and more like clickbait sensationalism. Very, very interested in the 5 journey through this paper. Yeah, I can see arguments, right? So before we even get into the paper, right, I want to say that in order to be as open-minded as possible, I kind of routed a way that I could believe this, but I don't think that this route exists, which is why I'm so skeptical, right? So if we take the assumption that we sleep because we have mitochondria, what proof would we... I'll actually turn this over to chat, right? I don't want to rob you of this opportunity, but for you, what kind of proof would you personally need to believe that the reason why we sleep is because of mitochondria? I'm not saying that mitochondria aren't involved in sleep. I'm sure they are. It just makes sense. But again, read that title and tell me that doesn't smell like sensationalism, right? I have an idea of what to rename the title in a more, in a less clickbaity kind of way that I would not be too upset over. But the thing about clickbait is that it kind of worked, right? I'm reading this paper now because of the title, even if I think that it is kind of dishonest. Hey, Astral, welcome, welcome. The mitochondria is EP. That is true. Upregulation of mitochondrial maintenance genes during sleep. So that would indicate that it has some sort of involvement, which I'm not against that kind of idea. Aren't there some rare parasitic eukaryotes that lost their mitochondria? We could try studying them. Oh, I did not know that. Are there parasitic eukaryotes? But do they sleep? That's kind of, I mean, maybe. That's actually, okay, so Seed, you've kind of taken where my mind wanted to go with regards to having an overall picture of the landscape of sleep. Whereas Clockwork, I think your comment is more supporting evidence that doesn't break the hypothesis, right? So I'm looking for ideas that would break this assumption that mitochondria are the reason why we sleep. That's kind of the line of inquiry that I want to go down. What site are you using to read this paper? I downloaded this as a PDF, but you can find the direct link to the Nature paper in the video description. It's open access, so you can definitely read along if you would like. Increased evidence of mitochondrial damage when sleep deprived, isolating the mitochondrial effects of sleep deprivation. That's also like, supporting and not necessarily trying to break this. So I think Seed's idea of maybe we'll try to find eukaryotes that don't have mitochondria and see what they're like, that's more in line of like, if you don't have mitochondria, are you not able to sleep? But this is kind of a problem, right? Because generally speaking, sleep is a function of having a nervous system, and having a nervous system is a function of being a eukaryote, and being a eukaryote is a function of having mitochondria. Right? Isn't that kind of circular in a way? This is almost a tautology, right? Every organism that has the potential to sleep has mitochondria, as far as I know, right? Seed, if you could find that species, that parasitic eukaryotic species that doesn't have mitochondria, I would love to learn more about that, because that would kind of fit in with where my mind is going. It's so weird watching a Fi stream while I'm in the lab. I've had Bobo put me on in the lab. It's kind of, if it's weird for you watching me in the lab, it's also weird to know that I'm being listened to in the lab. I do appreciate it though. Yeah, but chat, does that kind of make sense? Like the level of proof that I'm looking for? There's a kind of circularity with this argument. Is there a reason your PNG tuber is so sweaty? This is a true to life representation of what I am like when I stream. I sweat a lot in I don't know, there's something about energetically speaking that is so metabolically active that I do tend to get a bit sweaty. But yeah, I'm looking for an argument that breaks this circular logic, right? Every organism that could sleep has mitochondria. So that's why I immediately was like, this is some clickbait. If my Westerns turn out bad, I'm blaming you. They won't. They won't. My aura of competent Westerns will spread to you. Don't worry. You'll have area of effect buff here. But yeah, so that's kind of my mentality going in, right? There's a circular logic, there's a tautology here that is very hard to argue against because we don't have counter evidence or even a way to test for counter evidence, right? This is one of those things. I think Charis, if you're still in chat, you had introduced me to this idea of like, I don't know what you had called it, but like, I don't know, Euler's laser sword or something. Please, Charis, if you're around, correct me. This idea that there are certain questions that are, oh, by the way, Charis is a philosophy VTuber who I collabed with on his channel, and it was a lot of fun. So if you want to go check him out, I believe he chatted earlier. But there's this idea that like that there might not be a reason to ask questions that have no reasonable way of being answered, and I feel like this is kind of one of those things where this isn't necessarily a testable hypothesis, and that makes me feel weird. Monocerocomonodeus is a genus of flagellate excavata belonging to the order, blah, blah, blah. Okay, so if they're a flagellate, I'm guessing they don't have a nervous system because that sounds like a single celled organism. So sleep isn't necessarily, our conception of sleep is maybe not the same vocabulary, right? Because I'm not against microorganisms resting, but I think sleep is very neurological in terms of the way I understand it, at least. Oh, Chairs, you're still around, so please correct me on what I'm referring to, right? Like something laser beam, laser sword, Newton's laser sword or something. I don't remember what you called it, but um, it was a very spicy idea, and I liked it. Okay, so without further ado, plenty of other eukaryotes that don't necessarily have sleep pressure. Yeah, but I'm more so they don't have sleep pressure, but they're also not exactly complex enough to be able to sleep, right? I'm looking for something that has a complex nervous system equivalent that no longer needs to sleep. Whereas like for example, dolphins are a pretty famous case, but they sleep one hemisphere of the brain at a time, right? Like they still need to sleep. And if you're a single-celled organism, even if you're a eukaryote, right? If you're a eukaryote and you don't sleep, it's likely because you don't have the tools to sleep, like because I don't view non-activity as being the same as sleep, because this paper is specifically using the term sleep, which is not the same as rest or inactivity, right? Sleep to me is a complex neurological procedure, and if you have a nervous system, you likely sleep, and if you have a nervous system, you likely have mitochondria, right? It's circular and annoying like that. Loricifera have nervous systems, but they don't have mitochondria. Okay, so that is more in line with what I am interested in. So, are there organisms? Loricifera. Okay. Okay. For these guys, so these guys look like small. Interesting. This is in the area of biology that I really don't know anything about. These little critters. But okay, so I think this is a more productive line of inquiry with regards to this question, right? So these guys don't have mitochondria. Do other animals in this area sleep, right? Like, is there a cousin of this animal that does sleep without mitochondria? Like trying to find counterexamples of this, because this organism doesn't look to be okay. I'm realizing this question might be more sophisticated than, okay, A, I gave it credit for, B, what the paper gave it credit for, because at what point in nervous system development do we start seeing a bona fide need to sleep? Like, are there a lot of examples of animals with nervous systems that don't sleep? I'm very curious to know, because then we can kind of make that argument. Like, if every organism in this layer of complexity that doesn't have mitochondria also doesn't sleep, we can then compare other animals in this layer of complexity to the ones that do have mitochondria and see whether or not they sleep. So yeah, I would love to know that. That would actually, I think, more impress upon me the truth of this statement. Yeah. What about jellyfish? They kind of border on not being a single organism in terms of people viewing jellyfish as like a colony of single cells. So coordinating a complex routine like sleep might be kind of hard. Yeah, pretty interesting, I think. Genetic diseases caused by mutation in mitochondria's disease are a way to test it. Do those diseases impact sleep? So that's more like, again, in the sense where I don't really think that breaks this hypothesis. It just kind of supports that mitochondria have a role in sleeping. So it's a little different, right? Like, I don't disagree with the idea that mitochondria are involved in sleep. I would actually be very surprised if they don't, but it's, do you require mitochondria to sleep? That's a tougher barrier to get over. And if it seems like that barrier is impossible to get over, that's kind of my point. That's why this title reads to me like clickbait. Yeah. What about benthic animals like corals? Yeah, same thing of like, at one point did sleep evolve? And do we, in that layer where sleep first evolved, do we see a mix of animals that do and don't sleep? And does that correlate with having or not having mitochondria? To me, that would be the most definitive proof for, or not proof, but evidence, right? Proof is a mathematical construct, right? Evidence, that's what I would be looking for. But reading this abstract, I still think this is going to be a very interesting paper, just not one that I think will be satisfying for this question. Okay. All right, I'll read this last one by Clockwork, and then we should really get into the paper, or Astral is going to start docking me in terms of words per minute. Okay, here's one. You find another vertebrate that potentially counteracts mitochondrial pathways. This paper talks about some species of birds have way more mitochondria per cell than mammals. That I think is closer to what I would like to see. So like, if you break mitochondria in a certain way, I guess that's actually not that different from Guy's suggestion of breaking the mitochondria, or like genetic mutations in the mitochondria affecting sleep. But that would benefit from it not being a mutation, that it naturally evolved to do something like that. That would be closer, but it's still, to me, like the peak of the evidence would be at one point did sleep evolve, and then is there a mix? And if there is a mix, is it consistent with this idea that mitochondria are required for sleep? Okay, let's get into the freaking paper. Let's start by reading the abstract. I'll say that if I didn't read the title, I would actually be really hyped to read this paper, because I think this is a wonderful abstract and a really exciting line of inquiry. I just don't like the title, and I don't like the last sentence of this abstract. So let's read it together. To gain, oh, by the way, how's my mic sound, by the way? My new room is a lot bouncier in terms of sound and vibration. I hope that it still sounds okay. Did I miss anything? No, we just started. We just talked a little bit about what I would need to believe that we sleep because we have mitochondria. Okay, sounds good? Great. To gain a comprehensive, unbiased perspective on molecular changes in the brain that may underlie the need for sleep, we have characterized the transcriptomes of single cells isolated from rested and sleep-deprived flies. Already, I kind of love this idea because flies are a really good model organism to study neuronal processes. They're one of the premier model organisms if you want to study the nervous system. So I love this. I love that they're using flies. I love that they're using flies also because I think it's kind of a miracle, but I don't think that we've ever covered a fly paper on the channel before. And this might be the first one, which is super, super exciting because, flies are amazing. So let me break down the jargon. There's transcriptomes of single cells isolated from rested and sleep-deprived flies. So what this paper does is that they're taking single cells from these flies' nervous systems, and they are extracting the juice of the cells one at a time, and then they are getting an understanding of all the different mRNAs that these cells are making. So mRNAs, messenger RNAs, these are the intermediate between DNA, which is the code of life, and proteins, which are the thing doers of life. You could think of them like that. So by getting a transcriptomic read of each individual cell, the researchers can find out what the cell is actually doing in any individual moment. All right. Okay, so here we report that transcripts upregulated after sleep deprivation in sleep-controlled neurons projecting to the dorsal fan-shaped body, but not ubiquitously in the brain, encode almost exclusively proteins with roles in mitochondrial respiration and ATP synthesis. So let me break that down. So transcripts, that refers to mRNA. So the type of mRNA that goes up after flies have been deprived of sleep, they code for proteins that are involved in mitochondria stuff. And specifically, this applies to a particular region of the brain known for sleep control. And so the rest of the brain that is not involved in sleep doesn't have this distinction, right? So if you are outside of the sleep region of the brain, your mitochondrial mRNA, mRNAs that regulate your mitochondria, they don't really change when you're sleep deprived. But in the sleep region, that's when the mitochondria get kind of wonky. Okay, these gene expression changes are accompanied by mitochondrial fragmentation, enhanced mitophagy, and an increase in the number of contacts between mitochondria and the endoplasmic reticulum, creating conduits for the replenishment of peroxidized lipids. So that's a bunch of words. I'm not entirely sure. I didn't read this paper beforehand. I'm not entirely sure what each of these individual processes are. Hopefully we'll learn more about them in the background of this paper. but the implication of replenishment of peroxidized lipids is that so peroxidized lipids, this is like an evidence of mitochondrial stress. If you didn't know, your mitochondria are essentially factories for energy, right? The powerhouse of the cell, yada yada yada. We all know and love them. But what is less appreciated and something that you don't get in your 101 biology classes that I think I will go over in great detail in my next video, whose topic I'm still keeping a surprise. But mitochondria are not just the powerhouse of the cell, they're more like old-timey coal factories, right? They are powerful, they generate a lot of energy, but they're also supremely dangerous. They create a lot of byproducts that actually are very harmful to your cellular environment. And so peroxidized lipids is essentially like fatty acids that have reacted with the hydrogen peroxide that is naturally made in your mitochondria. So if you have stressed mitochondria, they're working really hard. And if you can't mitigate that damage, you end up with peroxidized lipids. So this is implying that, you have increased need to repair the things in the mitochondria, I believe. We recently did a transcriptomic analysis in my group. As a part of that, we also looked at some interesting effects on microRNAs. That's really exciting. I think transcriptomics is really, really cool and interesting, although it cannot replace actual hypothesis-driven work, right? It is an accompaniment to more experimental science. But I love that kind of thing. I think that's so cool. The morphological changes, so the changes of the shape of the mitochondria, are reversible after recovery sleep and blunted by the installation of an electron overflow. That's really cool. I don't know what that means. Hopefully we will find out what that means. Installation of an electron overflow, this is very engineering talk, and I'm hoping we'll get to see what that actually is. So apparently mitochondria change shape when you are sleep deprived, and then when you sleep, those mitochondria return back to normal. In the respiratory chain, inducing or preventing mitochondrial fission or fusion. It's a little bit of a grammar issue right there. Right, capitalized in. Okay, weird. Inducing or preventing mitochondrial fission or fusion in this region of the brain that controls sleep, alters sleep, and the electrical properties of sleep-controlled cells in opposite directions. Hyperfused mitochondria increase, whereas fragmented mitochondria decrease. Neuronal excitability and sleep. ATP concentration in this region of the brain rise after enforced waking because of diminished ATP consumption during arousal-mediated inhibition of these neurons, which augments their mitochondrial electron leak. Consistent with this view, uncoupling electron flux from ATP synthesis relieves the pressure to sleep while exacerbating mismatches between electron supply and ATP demand by powering ATP synthesis with a light-driven proton pump precipitates sleep. Okay, so here, let me take a moment to rack my brain for how to explain this. So electron flux, this is something that the mitochondria... Oh, by the way, I can't imagine that you're in my chat and you don't know who Clockwork is, but Clockwork, great friend of the channel, has a whole video on ATP synthesis and the mitochondria, which is amazing. And if you're getting kind of lost in the sauce here, go watch that video and you'll be in it, right? You'll be totally on board with what is going on here. But essentially, mitochondria use an electron pump in order to make ATP synthesis. And so what this is saying is that when you decouple that electron movement from, so I don't know how they're doing this, but they're going to separate the movement of electrons from making ATP. And when you do that, the organism no longer needs to sleep. That's okay. That is pretty cool. And that to me is getting closer to this idea that mitochondria drive sleep, because if you replace the function of the mitochondria in that targeted of a way, I haven't really thought about it all that much. I'm giving my kind of emotional reaction. Like emotionally, I'm more on board. And it's important to acknowledge the role that emotions play when you are doing some thinking, right? So I'm not putting any deep weight behind that, but if they could prove that if you tweak the mitochondria in just this way, where you are removing like the because you could argue that, okay, maybe sleep is because you don't have enough ATP. But if you do have enough ATP, I don't know how they're going to do that for these flies. If you do have enough ATP, but you remove one of the mitochondria's function upstream of that ATP synthesis, and then that's what's actually causing like sleep to happen, I'm much more on board now. This is different than a mutation in the mitochondria in that, okay, if the mitochondria are broken, you know, there could be 1,000 different steps down the line that could be affected that, you know, coincide with the ability to sleep. But this is like a surgical manipulation that leaves a lot of the surrounding tissue intact. So if you break the mitochondria, but you don't break their ultimate goal for the cell, which is to produce ATP, then I'm more on board, I think. More on board. Not going to say totally on board, but more on board for sure. Okay. I'm going to take a moment to read some chat. Can I sue my job for organelle injury? You can sue your mitochondria maybe, because they are not easy to keep around. Very, very tough. The mitochondria is the site of a lot of important metabolic pathways, as I understand it, though ATP is the most famous and rather essential. Yeah, there's a lot of other things that the mitochondria does. And that's why I'm not like throwing my entire weight behind this right now, because it kind of depends on how they're breaking electron flux. Because well, Clockwork might know more about this than I do, because, you know, he's more into the mitochondria than I am, but I am wondering if they could only decouple electron flux from ATP synthesis and keep everything else intact. I would be a lot more excited about the conclusions of this paper, I would say. What, do they hand pump protons in the mitochondrial lumen? I don't know. Well, we're going to find out. I'm super excited to see. Just getting into respiration processes in the mitochondria, feeling like a clueless intern. It's tough. It's tough. This is a whole like chunk of intro biochem course. And it's pretty nasty stuff to get into, but once you learn it, it's really cool. Okay. Now I'm scratching my head real hard over here. Kept myself in the dark in this paper so I wouldn't get spoiled. Oh, very fun. So we'll find out together. Okay, let's read this paper for real. So this is the introduction. Oh, this is a weirdly formatted paper, huh? Because we're already, you know, there's no intro thing like title here. The intro is one paragraph and then we're already into data, or is this more intro? This is a weird format. Or is this a review? No, this is a paper, right? This is surely a paper. Okay, whatever. Yeah, this is an article. That's such a weird. Okay, this is the shortest intro to a paper I've ever seen. That's kind of crazy. It just starts. Yeah, I was so surprised. I'm like, did I skip the intro? How are we already in the results section? That's nuts. Okay, intro. Sleep pressure, the process variable in sleep homeostasis, has lacked a physical interpretation. I agree with that. I'm not a sleep researcher, but I can imagine something as complex as sleep is not easy to detail. Although prolonged waking is associated with numerous changes in the brain of neuronal firing patterns, the strength of synaptic connections, the organization of subcellular compartments, metabolic concentrations, metabolic and gene expression programs, it remains generally indeterminable whether these changes are causes or consequences of a growing need for sleep. I don't know enough about sleep to back that generalization up. However, I will say that in my graduate studies, I did read a couple papers based on this idea that sleep is that when you sleep, okay, so I kind of like how this is worded because it is pretty careful. That when you sleep, you have neuronal rewiring and synaptic connections being rewired and homeostasis stuff going on. And I think it is the correct move to be so careful to say, is this because sleep does this, or is this driving sleep? I think that's okay to be that careful, and it helps for their argument here too. Okay. So let's see, where was I? Perhaps the only opportunity for separating causation from correlation. Yeah, exactly, right? So they were very careful, because I assume that when you sleep, you're doing rewiring, and that helps you learn and stuff, which is what I was taught, and what I've, with my own eyes read the evidence determined. But this is saying that just because these two processes happen at the same time doesn't necessarily mean that one causes the other. Maybe the causality arrow is reversed, which again goes to show you, right? Even if you have a lot of education, correlation and causation still is something that you have to be very on top of every single time. So points for the paper, right? I didn't think about that. So that's pretty cool. Exists in specialist neurons with active roles in the induction and maintenance of sleep. In these cells, sleep's proximate and maybe also its ultimate. You don't have to do this. You don't have to do this, right? In these cells, sleep's proximate causes and maybe also its ultimate. Like, this to me feels very immature. Like, this last sentence of the abstract, sleep like aging may be an inescapable consequence of aerobic metabolism. This is very clickbait, and I don't like it. I don't enjoy that they are putting words in my brain, right? This is forcing you to drink their Kool-Aid, as my advisor used to say, right? I don't have an issue with this. Sleep's proximate causes must interlock with the processes that regulate spiking. That's not controversial, right? The thing that causes sleep should be involved in brain activity. That's so not controversial. But also, if you put ultimate cause, it's like, that's ruling out a lot of other things, and you are trying to funnel us manually into your hypothesis, and that just gives me the ick. To delineate the molecular determinants of these processes in as complete and unbiased of a Am I being bad? Am I being a little too snarky? We are doing a very complete and unbiased experiment, but also our language indicates that we have a conclusion that we want you to believe instead of you making that conclusion yourself. Introduction jump scare, exactly. It's a single paragraph. It's so quick. As possible, we collected single-cell transcriptomics of the just because you do transcriptomics doesn't mean that you are doing an experiment in an unbiased manner. Have some money, Mr. Fly. Thank you so much, Cat Bertus. Thank you so much. That's awesome. Let the snark flow, embrace the dark side. We have cookies. I don't want to be the snark science VTuber, right? Like, leave that for other people to do. As I have said multiple times on my channel, I want to be a bard for biology. I want to espouse the glory and beauty of this field, and I actively don't want to crap on papers. But sometimes there's something on your feed that you just have to talk about, right? You got to take the good with the bad, but I don't want to be known for paper snark. Okay. Of the brain and rest of sleep-deprived flies, an encodable fluorescent marker allows us to identify and enrich for 24. Okay, so to explain myself on the transcriptomics thing, right? Transcriptomics allows you to take a snapshot of the whole cell, and so that data is unbiased in the sense where you're gathering everything from that cell. That doesn't mean you can't use that data in biased ways, right? So the idea that, this is more of a language issue, right? I'm trying to give maximum credit to the paper because I want the paper to be good, but the choices in the actual words is producing an emotional reaction in me that is negative. So I'm not criticizing their science. I'm criticizing their communication. Because I think, it's a Nature paper. This is going to be probably some pretty good science. It's just, I wish they didn't try to stuff their words in my brain. That is not as fun for me. Okay. An encodable fluorescent marker allows us to identify and enrich for 24 sleep-inducing neurons with projections to the dorsal fan-shaped body of the central complex and compare their transcriptomics response to sleep loss with that of other identical cell types. So this is saying we are going to genetically modify these flies to fluoresce in specific neurons, and then we'll figure out what they do. We can look at specifically those neurons with this technology. Okay. Maybe sus, but what if the authors find a way to earn the right to use this language? Yeah, I mean, they could. Hold on. I don't know what country these authors are from. Whenever I criticize language, I also have to be kind of careful about, is this English as a second language kind of thing? Am I being too hard? I actually can't really tell. I don't recognize what country these last names come from. And even so, like just because these last names are from a certain region doesn't necessarily mean that they aren't in an English speaking country. Um, but yeah, I think the term unbiased is often used in terms of describing uh, a certain type of data collection instead of a certain approach, and that's kind of what I'm brushing against here. Okay, they're all working out of Oxford as far as I can tell. Okay, oh yeah, Oxford down here. Yeah, exactly. Okay, so they maybe should have had like some eyes that could really change this one sentence, which is really annoying. And if you're thinking, wow, is every scientist like very anal about single sentences like this? The answer is yes. If you attend a journal club, there are often sentences like this that just make the entire room's eyebrows like go up. And it goes to show how important it is to learn how to write very well and to communicate very well. Because if you are writing for a skeptical audience, even one sentence like this can put everybody on like a good mood to a very defensive mood. And I'm feeling pretty defensive. Oxford lab, but based in Switzerland. Okay, so I mean, I don't know how extensive English language learning is in Switzerland, but yeah, I maybe can't go that hard. I don't know how hard I can go, but a little bit from the Oxford side, y'all should have maybe thought about wording this differently. Okay, let's get into the results. Traces of sleep loss in mitochondria and synapses. Okay, so this title indicates that we are not looking at causative data, which is fine. This is like established in the context. So when you lose sleep, we're going to be looking at some cellular evidence for that. Like, what does a sleep deprived mitochondria look like? What does a sleep deprived neuron look like? That's what this section is going to be about. Drosophila brains were dissociated into single cell suspensions. Oh, that's got to be. I always find that people who do this to be so incredible because I've done this for a mouse brain, but mice brains are huge compared to fly brains. Like the technical ability needed to do this has to be insane. Neurons expressing GFP. This is a fluorescent protein, green fluorescent protein under the control of some Gal4 promoter. Okay, kind of annoying. This Gal4 is one that is often used in Drosophila. I'm not a Drosophila scientist, so I actually don't know what this is, but I'm going to quickly Google this. So R23E10, what is this? What is this promoter? Okay, so this is a promoter active in specifically sleep promoting neurons. Okay, so that's what that is. Okay, that makes sense, right? So they are labeling only the sleep neurons with GFP. And then they were isolated by flow cytometry. So this is a method by which you can separate a mash of cells into single cells, and then you launch them through a detector, and then that detector will measure the amount of fluorescence for each cell. It's an incredibly powerful technology, very, very tricky to get to work properly. The machine clogs all the time. But essentially, they are saying we're going to look at every single sleep neuron and check their fluorescence. We performed single cell RNA sequencing on cells contained in the GFP positive and GFP negative fractions. Okay, so this is saying we take all the fly neurons, and every time we get a neuron that fluoresces, we bin that into its own category as a neuron that is sleep important. And then we are going to take those separated single cells, suck the juices out of each individual cell, and see what RNAs those cells are making. About 45% can speak it well enough to get by. Okay, yeah, I don't want to be, again, too hard on the Switzerland side of things, but there's like no excuse if you're in the Oxford half of this collaboration. Okay. Nominated by this, were identified by the expression of GFP, the neuronal markers, embryonic lethal, abnormal vision. Okay, so this is all like Drosophila. I think this is maybe a Drosophila specific marker because I don't recognize this from my studies in mice. But neural synaptobrevin, I believe this is a pretty universal neuron marker for synapses. and allostatin. I don't actually know what this one is, but essentially they're taking a look at the cells that fluoresce, and they also have to have stuff that shows that they are neurons, whose transcriptional enhancer provides the genomic fragment driving the expression in these lines. Okay, sure. Most neurons in this domain contain machinery for synthesis and release of either glutamate or GABA. So these are neurons. Some also, so these are specific types of neurons, the GABA, glutamatergic and GABAergic neurons. Okay. Some also transcribe the genes encoding for the vesicular acetylcholine transporter, a common occurrence in non-cholinergic neurons, which rely on a microRNA to suppress its translation. Indeed, fluorescent sensors detect the release of glutamate, but not of acetylcholine from these cells. Okay, so this is basically saying we took all the cells that we engineered to fluoresce in that sleep important sector, and we found out what other mRNAs they make, and they make glutamate and GABA. These are two neurotransmitters stuff. So this is essentially saying, okay, the neurons that we think are important for sleep that we isolated, they are in fact neurons. They do in fact do neuron things. And they do neuron things in the way that we expect them to. So this looks like a win for them. I'm not an expert in these experiments, but I'm just going to take their word for it. Okay. In the Drosophila central nervous system, glutamate and GABA both act on ligand-gated chloride channels consistent with the known inhibitory effects of this sleep region on their postsynaptic partners. Okay, yeah, again, just we know that the cells important for sleep are glutamatergic and GABAergic, and that's what we found, so we pat ourselves on the back. That's cool. Members of this pattern that signal via one or the other of these inhibitory transmitters are anatomically segregated. A pair of GABAergic cells target the suboesophageal ganglion, sure, while a larger glutamatergic population innervates the dorsal fan-shaped body. Okay. This latter group of bona fide cells formed a distinct gene expression cluster of 323 cells amongst all glutamatergic neurons in the brain. This cluster was defined by genes collectively known as these enhancers, and encoding the known blah blah blah, such as Rho-GTPase. Sure, this is an actin cytoskeleton regulating thing that's also important for synapses. It's like a lot of words, but I'm not entirely sure what their point is. I think the entire point of this, and this is like, I'm not going to get a whole lot from this because this is talking very specifically about the cellular anatomy of the Drosophila brain in this specific region that I'm not very familiar with. But I believe all this is saying is that, yes, the neurons that we're looking for are in fact the neurons that we have isolated. Okay, here is the part that is actually interesting here. Okay, so gene ontology analysis. So yeah, all that was to say we isolated the correct neurons. Now we're going to see what those neurons are making. So gene ontology analyses of the 122 transcripts whose levels in this sleep region changed after 12 hours of overnight sleep deprivation pointed to only two sleep need dependent processes: mitochondrial energy metabolism and synaptic transmission. So that is figure 1C. I'm not going to look at the supplemental, otherwise we'd be here literally all day. So let's look at the data. Okay, so let's see here. So this is what flow cytometry data looks like. Uniform manifold approximation and projection UMAP representation of glutamatergic neurons according to their gene expression profile. So I'm not like a flow cytometry guy. So I had to teach myself how this works every single time. But essentially, if you want a very casual explanation of what we're looking at here, UMAP analysis is a way to visually separate the characteristics of these cells into distinct categories. So you can kind of see that there are these unique clusters. And when we zoom into these clusters, these are distinct populations within these, right? So there are these like gray cells up here. These are like lumped together in a certain axis. But then there's this unique cluster over here. And when you take a look at this cluster, you find that within these clusters, there's a shift between when the neurons are rested in teal or a blue, and then when they're sleep deprived, the characteristics shift in a kind of notable way. So if there was no change between these neurons when they were resting and when they were not resting, we would see the magenta and the blue completely overlap, but it looks like there's some change. It actually doesn't look like a crazy change to me, but it's something, I think. Thank you for becoming a progenitor cell, Stephen. Thank you, thank you. Thank you so much. Okay, so what does B say? Log normalized expression levels of these markers. Okay, we're going to ignore that. Volcano plot of sleep history dependent gene expression changes. Ooh, volcano plot. Okay, so log fold change after sleep deprivation on this axis, and then Bonferroni corrected P. So I wish Mylan were here. I'm a little rusty looking at these kind of plots. I think the p-value as it goes up, these are an indication of how statistically significant the data is. So the higher on this axis, the more statistically sure we are that these differences are real when you are sleep deprived versus not sleep deprived. Okay. And then on the x-axis, we have the log fold change. So either you are increasing or you are decreasing. And so the most tempting genes, these are mRNA transcripts, the most tempting genes are going to be the ones that are kind of in this upper quadrant. And then it looks like they highlighted different genes that are important for the mitochondria, right? So it looks like subunit of mitochondrial complex 1, 2, 3, 4, and 5. It looks like a lot of the genes that are, let's see, upregulated, that go up, are mitochondrial genes. But I mean, if you look at the number of genes that aren't mitochondrial, I mean, it's quite a bit, right? A lot of gray stuff down here. Although I think maybe these grays are where they cut off their significance. So maybe the important ones to look at are black and then the colored ones. So there's a lot of black here that don't correspond to mitochondrial complexes. So just keep that in mind. This would have been the amount of presence of various RNA transcripts as separate dimensions. Yeah. UMAP is a matter to attempt to approximate this high dimensional data to a page. Yes. That's an excellent explanation, Marlon. So this clustering is like looking at the characteristics of their RNA expressions. And because that is very difficult and messy, UMAP is a way to take that messy data that is very hard to imagine and compresses it down to a 2D image. And then you can kind of see if they cluster, they're more likely to be a part of the same group. And if they don't cluster, then they're likely to be different populations. Thank you for that. That's a really good explanation. That kind of jolted my my memory. But yeah, so a lot of these proteins that are involved with sleep, as we can tell by the difference in sleep deprived and sleep rested flies, are mitochondrial proteins, although they're not all mitochondrial proteins, right? But when we look at the what is it called? The the gene ontology, the enrichment. So you take these genes, and then there's a database online that can lump these into general categories for you. So let's take a look at what these categories are. So this is fold enrichment. So over here, we have genes that are downregulated. And then up here, we have genes that are upregulated after sleep deprivation. So let's take a look at what these categories are. So we have synaptic target attraction. I actually don't know what that means. We have mitochondrial electron transport, cytochrome C to oxygen. So that's pretty huge, right? The category of genes that is the most upregulated after sleep deprivation is stuff having to do with mitochondria and electron transport, which is good for their hypothesis. Synaptic target attraction, don't know what that is. Glycolytic processes, so this is metabolism. And this is kind of, not mitochondria specific, right? Like glycolysis feeds into mitochondria, but you can do glycolysis without mitochondria. So this is kind of just another note to make that mitochondria are not the only thing affected by sleep deprivation. Proton motive force driven ATP synthase. So this is synthesis. This is what Clockwork made his video on, the wonderful enzyme known as ATP synthase. So genes regulating that are upregulated when you are sleep deprived. Aerobic electron transport chain. Again, we're going to see a lot of these repeat categories. I'm sure that they overlap in some way as well. mitochondrial ATP synthase coupled transport, ATP biosynthetic processes, oxidative phosphorylation, ATP synthesis coupled transport, pyruvate metabolic processes, respiratory electron transport chain. Okay, so this is cool. Okay, and I gotta give the authors a lot of credit for this. This is probably what they meant by everything that came up was mitochondria related, because if we take a look at 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, all like 10 out of 10 of these highly upregulated genes are involved in the mitochondria. Now, this is not complete. That's important to know, right? Okay, hold on. Let me reread this. Gene ontology, 122 transcripts changed. Sleep loss, all of this stuff, downregulated. Yeah, okay. So of the 122 transcripts, these are the major categories that they were split into. So whether or not there are more categories, I'm not too sure of. But if they do exist as categories, like below these top 10, they might not be all that significant. Although, given their language, I'm unsure, right? Because I'm put in the skeptical mode, I don't know what's below this, because this is 25-fold enrichment. What about categories that are like 10-fold enrichment? Just because something is super highly enriched doesn't always mean that it's super important. And, you know, you can think of something like if a gene transcript is very tightly controlled, even a 1.2-fold increase could be very significant. So because I'm in a bit of a, you know, skeptical mood, I would probably rather, like, I don't know whether or not there are other categories that might be worth looking at that just didn't make this insane, like 25-fold enrichment cutoff here. Yeah. There's so many things they managed to lock down here. Geez, how do you even separate signal from noise in data like this? Any chance some of these are outliers? I don't have the expertise to really go into that. It's a lot of computational biology, although I'm sure Resta might be able to do it. Resta being someone I've collabed before, who is a, she's not a computational biologist, but I forget what her title actually was. Bioinformaticist, I think. Yeah, they have skills that I don't have. Let's take a look at the genes that are downregulated during sleep deprivation. So synaptic target attraction, this is not a mitochondria thing. Calcium regulated exocytosis of neurotransmitter, also not a mitochondria thing. Positive chemotaxis, that's very weird. Yeah, that's weird. Positive chemotaxis. I know that during development, neurons crawl, like when the brain is developing, there's like a whole regulatory thing where neurons will crawl down a certain pathway to get to their final destination. But what the hell is chemotaxing? So for those of you who don't know, chemotaxis is a cellular process by which a cell can literally follow a chemical signal. If you know anything about neutrophils, neutrophils are premier chemotaxis enjoyers. Neutrophils can smell signs of infection and follow that signal. But if we're talking about neurons, what the hell is chemotaxing? I think that brings up how big their sample size is. How many times they ran this experiment? Do they give those details anywhere? That is in the paper, I think. I think it's like 300 something neurons. I don't know from how many brains, though. In my training, I was always led to believe that the number of neurons is less important than the number of brains, right? Because there are technical replicates and there are biological replicates. Technical replicates would be ends. You would base them, yeah, 200 something cells, right? But you don't mention how many brains. Okay, so this is a really good point, I think, Stephen, that you brought up. While an N of 200 cells seems very impressive, if they all come from one brain, that's a biological replicate of 1. So this might be buried in their methods. Okay, maybe we should tab over to the methods to check how many brains were used. Because here's the thing about 237 cells gathered from one brain. Yeah, okay, well, because I'm not a transcriptomics expert, so I don't know what the convention, I don't have it memorized what the convention for this analysis would be. But the problem with this is that if these are all from one brain, then that means that everything that correlates in that one organism is also affecting everything in this data, right? You know, one Drosophila is just genetically a little weird, ate a little more food or ate a little less food during development, and now that little bit of experimental difference can affect entire networks in their direction. Yeah, let's go over to the methods. Two brains are better than one. Exactly right, A. That is exactly right. So let's tab down to the methods and see. Oh, let's see. God, this is also difficult to read. Sleep assays, sleep deprivation. I have read the methods first? Yeah, probably, but I'm not going to be able to get a lot out of it because I'm not a fly researcher anyway. I think it's okay to look at the methods as we need to, at least for content purposes. Okay, so this is how they did sleep. Brain dissection. On each experimental day, averages of 186 rested and 144 sleep-deprived female flies were retrieved alive and dissected. Is that for the... Okay, differential gene expression. Here we go. Gene ontology. All the way down here. They didn't do it in chronological order? Okay, that's fine, I guess. Differentially expressed genes were identified by this, using RNA assay counts of two comparison groups. Gene ontology, database. Didn't say how many brains were used. I don't want to come down too hard on this because again, I've never done one of these myself, so I can't be too sure. But I'm like relatively sure you shouldn't be using just one brain for this and then making conclusion based off of one brain. That's really annoying. Yeah. I don't know, Lunkel, if you're still around, if you can offer some insight having actually done transcriptomics analysis. If you can get an insight into how many brains were actually collected, because I don't know. Okay, I'm gonna go take an idle break, a scientist break. I'm gonna play an ad. Didn't they just say 144 female flies were dissected? I think that was. Hold on. Am I being too mean for no reason? This is for the scramble RNA-seq, so this is a different experiment. And I can't find any other mention of 144. Yeah, this is specifically for cell collection for scramble RNA-seq, which is different than differential gene expression. Right? I believe these are different experiments. Maybe they're not. I didn't see anything about scramble RNA-seq. Okay. I take everything back. We're chilling here. Sorry. scRNA, I read it to be scramble RNA, which is different. scRNA here refers to single cell RNA. Okay, I apologize, paper authors. I'm not as mad. They used 144 brains, and that totaled to this number of cells. Much better. Why didn't you put it in the caption though? You put the ends. We just should have added they came from quite a few brains. So I'm a lot happier with that. I'm a lot happier with that. That's not crazy to me. Yeah. Okay. Very, very cool. Okay. Yeah, I'm gonna go play an ad and I will be right back. Don't go anywhere. I will see you again very, very soon. All right, I'm back. Did you like the waiting screen? don't get to uh don't get to use that too often. Are we back from the ad? Is anyone still watching an ad? I'm gonna go ahead and just check the chat. I know that when I choose to deploy the ad, not everybody will get the ad, which I think is kind of wild for a stream. I mean, like to have, because that kind of makes taking breaks kind of inconvenient. But I'm guessing if no one is saying that they are still in the ad, then I can continue to go on. Okay, so I will point out that because this paper was worded in a certain way, I was automatically put in a bad mood reading the paper. They actually did this part fine. They used a ton of brains. And I guess that each brain doesn't have that many of these sleep neurons, which is why it takes 144 brains to get like 300 cells or whatever. So that kind of makes sense to me. So to kind of recap, in sleep deprived neurons, you get a massive upregulation of a lot of genes that are important for the mitochondria, and then the downregulation of a bunch of genes that don't actually seem to be that important for mitochondria. So it's kind of interesting. Things that go up are mitochondria during deprivation, but things that go down during sleep deprivation are just like normal neuron stuff, which is kind of nuts. I just got here. I was my mitochondria didn't let me get here sooner. Very fair. Blame everything on the mitochondria. Okay. Yeah. Oh, welcome, Fire Eyes. Welcome, welcome. Okay. So let's continue. So I do think that that's interesting, right? Sleep deprivation specifically seems to upregulate mitochondria stuff, in general, and maybe some other things, but they're not listed on the group category. Certainly, they are the most impactful on the mitochondria in terms of gene expression, right? There's a very important distinction to make that we are just looking at mRNA and not at protein or function. Okay, but I'm guessing that's where we're going to soon. Okay, so we have 12 hours of overnight sleep deprivation. I want to know how they are depriving sleep to Drosophila. That seems like something that we should read now before we get too much deeper into the paper. I don't know if you can hear, but Perkinji is unhappy. Perkinji, you can come out and hang out in the streaming room. You don't have to just meow sadly in the living room. Okay. Sleep deprivation. In standard sleep assays, females aged 2 to 4. Okay, so already I'm a little confused as to why we are only collecting data from female Drosophila. This is something that maybe a Drosophila scientist could pitch in with, but to me, as a non-Drosophila person, I don't know why we are not mixing sexes, because now again we have this correlative thing where are mitochondria only important for sleep in females? Like there's just, I don't know, it's a little annoying, but I'm sure Drosophila scientists might have a good reason to say that. Individually inserted into these glass tubes containing food reservoirs loaded into the Trikinetics Drosophila activity monitor system and housed under 12 hours of light, 12 hours of dark conditions at 25 Celsius and 60% relative humidity. Flies were allowed to habituate for 1 day before sleep, classified as periods of inactivity lasting longer than 5 minutes. Sleep and circadian analysis MATLAB program. Yo, MATLAB. Were averaged over 2 consecutive recording days. Immobile flies, fewer than 2 beam breaks per 24 hours, were manually excluded. Our standard method of sleep deprivation used the sleep nullifying apparatus, a spring-loaded platform stacked with Trikinetics monitors, was slowly tilted by an electric motor, released and allowed to snap back into its original position. This mechanical cycle lasted 10 seconds and repeated continuously for 12 hours, beginning at Zeitberger. My Europeans in chat. Any idea what this word means? Beginning at Zeitgeber. Time 12. Flies expressing TRPA1 in these sleep neurons were mechanically sleep deprived at 29 degrees Celsius. Okay, weird. We're gonna have to look up what that one is later. Time giver. Okay. Interesting. Anything that synchronizes the circadian clock. Okay, this is some sleep specific jargon that I'm going to just accept. But essentially, the way that they are depriving the Drosophila of sleep is by not allowing them a stable platform to rest on. It's like if your bed like spun every 10 seconds or something. Um, I see. So okay? So this term is saying we are determining a time. We are calling it 12, and then we are rotating based on this arbitrary time that we set. I believe. Uh, it's a Zeitberger and not a Zeitgerber. I don't know. I don't know. Rhythmically occurring natural phenomena which acts as a cue and regulation of the body's circadian rhythm. So circadian rhythm time 12. Okay, I'm guessing this is jargon specific, arbitrary. We begin at this time and then we repeat it every so often. Okay, so without being a sleep researcher in Drosophila, I have a little bit of skepticism with regards to this. And I don't, and chat, you can kind of help me figure out if this is something worth being considerate of. That like mechanical perturbation of sleep has its own correlates, right? It's stressful, right? In a way that might be a little different than just having like a 24-hour light on, if that makes sense. Or a loud noise. Like I guess these are all. I guess you might not be able to separate a method of sleep deprivation from physical stress. And why do I say that? Is because if I would not be surprised, for example, that your mitochondria would be more active if I were to jump scare you. Right? These Drosophila are essentially getting jump scared, right? Because the electric motor is tilted and then snaps back to its original position. So there's this jolt that happens. And you can imagine that if you're being shooketh, like if you're being jolted, then that's probably not just sleep deprivation. That's probably also the physical stress of being jump scared, right? And if if Drosophila have a fight or flight, that could be a correlate, right? A confounding variable that is maybe contributing to the data, right? So that is why we read methods, right? I was curious to see how they were going to do this. And I'm not saying this kills this experiment, right? Because there might not be a good way to deprive someone of sleep other than to shake the bed that they are in, at least for flies. For humans, you can play a loud noise, which is also stressful, right? Things that are sleep depriving are also stressful. So there might really not be a better way to separate the two here, but we do have to acknowledge that there might be some more things to have to untangle later into the future. I don't know. When I don't get sleep, I'm pretty stressed. Tend to extend that state to anyone with the misfortune to interact with me. Yeah, exactly. Like there might not be a way to separate sleep from the induction of stress. Although sleep causing stress is like different, right? This is there. Ideally, there should be a clean way to deprive Drosophila of sleep without physically stressing them. Galea, I don't understand. So what we're talking about is this paper that is purporting to claim that the reason why we sleep is because we have mitochondria. So we are going into deep detail into the paper about whether or not we believe that that is true. Okay, so that is but in the next paragraph they actually address that. Okay, that's what I like to see. That's what I like to see, right? I was just saying how I was worried that physical perturbation of Drosophila might be causing them stress, which could be affecting their mitochondria that is not related to sleep. And apparently they have other methods. Oh, that's very exciting. See, I'm not mean. If you do good, I am very happy to read good science, right? To guard against potential side effects of regular mechanical agitation, we explore two alternate methods of sleep deprivation with comparable results. In some behavioral experiments, in their supplemental, a vortex mixer stacked with these monitors produced horizontal circular motion stimuli with a radius of about 1 centimeter at 25 hertz for 2 seconds, in contrast to the rhythmic displacement of flies in the sleep nullifying apparatus. Okay, so I'm guessing this means that they put the Drosophila on like a carousel, which is kind of cute. And then in order to prevent them from getting used to the carousel, they were randomly disturbed. Um, like the carousel would randomly go on and off, which I think is more gentle than being like shaken awake every once in a while. It's still maybe stressful, but I would guess that this is more gentle than mechanical agitation. In some morphometric experiments, flies expressed, we're going to get into what this is later because I don't know what this is right now, which produced sleep deprivation without sensory stimulation. Okay, so this is probably a Drosophila that was genetically modified to be bad at sleep when when certain conditions are met, which is very exciting because then I get to talk about genetic engineering, which I think is very cool. Would you be able to feed them amphetamines to keep them awake without physical agitation? I think that is also flawed in the sense where now you're dealing with metabolism and stuff like that. But if like if you get the same conclusion, then that's a good experiment. If you don't get the same conclusion, then that experiment doesn't actually teach you anything, which makes it okay. Like on the tier list of experiments to do, I think that lands itself in it would be cool to have seen, but not necessarily informative if you get the results that so like there's like a mental tier list I have for experiments where the best S tier experiments are informative no matter what result you get, and those are always the experiments that you want to design around. It's not always possible depending on your system. And then below that are experiments like Fire Ice, what you're suggesting, something that could be informative if you get the right result, but then not informative if you get the wrong result, right? So I think it's not a bad idea. It's just not the best like answer. It might be the best practical answer, but it's not the best ideal answer, if that makes sense. I do like that experiment. Or surgical intervention to remove the ability to sleep. I believe that would be a little worse than Fire Ice's recommendation, just because once you get into surgical intervention, then you have to deal with inflammation. And as someone who is a neurobiologist with a focus on the immune system, I really don't want to touch anything having to do with the fly's immune system with regards to their sleep, because then it's like, okay, if they have mitochondrial stress, is that because everything is stressed because you perform surgery? Not entirely clear. It's the same form as Fire Ice, like in terms of like if you get the right result, it's good. If you get the wrong result, it's meaningless. But I think it's more likely that an amphetamine or something, a pharmacological intervention might be a little gentler than surgery. But it's not a bad idea. Okay. So I'm looking forward to learning what this trip A1 Drosophila is, because I'm guessing that'll allow us to see why Drosophila are such a powerful model organism for genetic manipulation. But we'll get there when we get there, I think. Okay, I am so low on the words per minute right now. That's because there's just so much to talk about in this paper. But that's okay. That's okay. I've given up my throne as the fastest paper cover. I want to take my time with this, of course. Um, okay, sleep loss led to the selective upregulation of mitochondria stuff, and then stuff that is involved with neuron stuff was downregulated during sleep deprivation. With the practical limits of our analysis, we cannot extend to every conceivable neuron type. This transcriptomic signature of sleep loss appeared unique in the sleep region of the brain. It was absent from two cell populations with comparable numeric representation in our data, namely projection neurons of the antennal lobe and Kenyan cells of the mushroom body. And it was also undetectable in a combined analysis of all of the non-neuronal sleep cells. Okay, so this is trying to make the argument that the mitochondrial upregulation is specific in the region that produces sleep. And then when you look at the other data, when you combine the data, this is pretty good, right? This is double checking to see that there's nothing else interesting happening in any other region of the brain, which is consistent with this hypothesis that mitochondria are driving sleep or involved in sleep. Are fruit flies sensitive to caffeine? I don't actually know. It's a good question. There are, however, subtle hints from an independent transcriptomic study that sleep history may alter the levels of mitochondrial components, not only in this region, but to back up, right? Fire Ice and Steven, your experiments are good ideas. However, I think with the length of this paper, I don't feel the need to do them right here, right now. Yeah, I think they maybe have done enough. Those are good future directions, but I don't want to mark that against these authors. Like, I don't think that's their fault. Eventually, you have to cut the paper somewhere, and I think that's a fair line to draw. That kind of stuff can be for something else, a different paper. Okay. There are, however, subtle hints from an independent transcriptomic study that sleep history may alter the level of mitochondrial components not only in this region, but also in R5 neurons of the ellipsoid body, another element of the sleep homeostat. Okay, good. Acknowledging that there is data out there that might suggest that there is larger involvement outside of their particular thing. But I don't think that this breaks them at all because um, it's still talking about mitochondria just in a different part of the brain. But I'm pretty happy with staying with this region of the brain for now. I wasn't saying it as marks against them or as a possible way to follow it up. Yeah, I figured you would. I just, for VOD watchers or for people listening, I want to be very, very careful every time I talk about a paper to not come off as someone who is overly critical because, you know, one of the things that I'm most afraid of is being canceled over some stuff that I was ignorant about, right? I'm very careful to only talk about what I am pretty confident in, and I want to give as much credit as I can because I don't want to attack science. So it's just an opportunity for me to remind the audience that, like, I am not out here looking to tear down the paper, but I am going to express my annoyance when my annoyance I think is warranted. The remainder of this article examines the causes and consequences of the differential expression of genes encoding mitochondrial proteins. Here, a parallel study investigates the role of presynaptic plasticity in the wider context of sleep need dependent. Okay, so they're pointing to another paper. It's kind of weirdly worded, but okay, that's okay. If I remember pharmacology correctly, caffeine promotes wakefulness by altering circadian rhythm. Amphetamine works via NE, so directly on neurons. And then I think maybe amphetamine would be better. Yeah, the closer we can get to affecting fewer things as possible, the better we can maybe tease apart the complicated web of causation and correlation. Okay, this is crazy. I don't know, Clockwork, if you're still around, but this title of this section is bonkers. Okay, I'm gonna let you take this in. What this section is about to claim: a mitochondrial electron surplus induces sleep. So this section is about to claim that if mitochondria, that the thing that makes Drosophila sleepy is pumping mitochondria full of too many electrons. Okay, that's a very tall order. That is a very tall order. That's a crazy thing to claim. So of course, something as crazy as this needs to have some crazy evidence behind it. So I'm very excited to see what the heck this is happening. I think my laptop is overheating. Had written a long yap about high word per minute isn't necessarily linked to a good stream, but then crashed and deleted it. Gonna go try to fix it. Have a nice stream. Thanks for stopping by, Astral. Yeah, I think the word per minute thing is mostly a meme to clown on Cider. That's, I think, mostly what that is. But I think we can all agree that her streams are very excellent, regardless of the words per minute. And as you can tell, I'm not very plussed about my own words per minute here. It's more of just a game that we all play with each other. Okay, especially because when the science is this interesting, I don't want to actually go fast. It's really, really good. Too many electrons equal ETC isn't being efficient enough moving them to oxygen. Claim that mitochondria become less efficient throughout the day. Yeah, I think that, like, in order to claim this, okay, I'll put my money where my mouth is, right? I think in order for me to be happy with this statement, you would have to pump the mitochondria full of electrons and then prevent. No, you know, I'm a back up. I'm gonna be generous. You would have to manually pump the mitochondria full of electrons somehow without affecting any other process that is not mitochondria related for me to be happy with this, right? And I say that very carefully, but I'm putting that out there so that I remain true to my values, right? If you are pumping mitochondria full of electrons by I don't know, like Clockwork, you can check me if this works. Like cyanide is an electron transport chain inhibitor. Maybe that causes too many electrons to build up. But then like cyanide is just overall like a poison. So that's something to consider. So how are you going to experimentally produce an electron surplus within the mitochondria without like just poisoning the animal? Like okay. If they can pull this off, I'm gonna be pogging, right? I'm gonna go crazy. Also, me and Eggy are making a Z thing. Can we use brief audio snippets from your vid stream? Yeah, of course. Absolutely. I gotta get some work done as well. Hope everyone has a great day and keep it bright. Thank you so much. Thanks for hanging around. I have to go work in 40 minutes, but my mitochondria is screaming at me, as they do with everybody. Thank you for stopping by before work. Oh, there were just some YouTube videos explaining methylene blue's effect on neurons/electrons. Maybe Drosophila will want some during this study. Maybe. This mechanism relies on a subunit called hyperkinetic, which is a subunit of a pump called shaker. Very cool name. I love like Drosophila name stuff. Okay, so how are they going to do this without messing a whole bunch of other stuff up? I am very curious to know. The prominence of mitochondrial components in the transcriptional response to this sleep-producing region in the brain to sleep deprivation offers just, you don't have to like mention this though, right? Like, offers unbiased support. It's just, it sounds weird. It just sounds like, you're trying too hard, man. Just let your data speak for itself. For the hypothesis that sleep and aerobic metabolism are fundamentally connected. This hypothesis gained a firm mechanistic footing. You know what? Okay, you know what if this were a student, I would take points off, because this is an interpretation, right? The language here, this, because unbiased is a word that you can use in this like big data experiments, I'm not as mad. This hypothesis gained a firm mechanistic footing within discovery. Like, this is too much. Firm message, like, just say there is evidence to support. This veers into the territory of casual writing. And you know, you could argue that maybe science papers should be written for the public in a more casual way than this. But this statement here is why papers have to be written as objectively as possible. Firm mechanistic footing is trying to create a reaction in your brain that makes it seem like there's a lot of evidence for this, right? But you don't have to say that. You just say there is evidence, or there have been multiple papers suggesting, right? And then you link those papers. But take a look at this. This hypothesis gained a firm mechanistic footing with the discovery that this regulates sleep through machinery that gears their sleep-inducing spike discharge to mitochondrial respiration. And then there's one citation. Firm mechanistic footing followed by one citation. It just feels bad, man. It feels so bad. I so far, I actually really like the science so far. Just the data, looking at just the data, I'm really enjoying it. But when you write like this, it just makes me feel bad. It's a paper that sometimes feels like it's trying to be a review paper. Exactly, Clockwork. Like, this is something that you could maybe get away with in a review paper, but don't, like, man, don't lower yourself to writing like this. That's my tip for everybody here. Don't debase yourself by trying to say something like this. This is trying too hard. Come on. The centerpiece of this mechanism is hyperkinetic, the beta subunit of the voltage gate potassium channel shaker, which regulates the electrical activity of this region. Hyperkinetic is an unusual aldyl-keto-reductase with a stably bound nicotinamine adenine dinucleotide phosphate cofactor whose oxidation state, NADPH or NAD+, reflects the fate of electrons entering the respiratory chain. Okay, here is where like my brain starts to spiral, because I'm not like a metabolism guy. So bear with me. I'm going to come at this from a much more casual view than some of the neuroscience stuff. Neurobiology stuff, I should say. So let me reread this. And okay, so I'm gonna read this a little silently to myself so I can really lock in here. Okay, so in Drosophila, in this region, they are claiming that metabolism and sleep are linked by this particular subunit of this particular potassium channel, which regulates the electrical activity. Okay, so this channel is not a metabolism channel. This channel is a voltage gated neuron protein. So this is a protein that regulates the polarization of the neuron, I'm guessing. This is something to do with electron firing. And then is an unusual aldo keto reductase. Unusual? Just say it's a reduct. Why put unusual here? I don't know. There's a lot of fluff in this paper that I don't understand. With a stably bound NADPH cofactor whose oxidation state reflects the fate of electrons entering the respiratory chain. Okay, so this is the connection, right? So shout out to my biochem students who have to learn glycolysis and electron transport chain. This is a neuron channel that has a spot for a metabolic, a metabolite, NADPH, right? So if you took biochem at some point, learned about glycolysis at some point, you would know that in glycolysis, you produce NADPH, which is a high energy electron carrier, which then gets shuttled to the mitochondria to do mitochondria things, right? So again, this is why you have to learn glycolysis, despite it being such a hassle. This is the link between the neuron protein, like the neuron side of things and the mitochondria side of things. So not only is this channel something to do with the electrical activity of the neuron, it is linked to the mitochondria by this cofactor that is produced during respiration. Okay, so that's interesting. I've never heard of a channel that links metabolism to neuron firing in this direct of a manner. Electron transport chain, the fun fair ride for electrons. Yeah, exactly. The the infinite carousel. So it can act as a signal of intercellular metabolism. Looks for signals from shaker. You can infer metabolic stress of lack thereof. I'm not entirely sure yet. I'm just trying to get a handle of what hyperkinetic and shaker actually are. Reflects the fate of electrons entering the respiratory transport chain. When the demands of ATP synthesis are high, the mass majority of electrons reach oxygen, and an enzymatic reaction catalyzed by cytochrome co-oxidase. Only a small minority leak prematurely from the upstream mobile carrier coenzyme Q, producing superoxide and other reactive oxygen species. Shout out to the next video that I'm making. You're going to hear a lot about superoxides, which I've talked about on the channel before, but I'm going to go into a lot of detail on the amazingness that is the mitochondria and the superoxide system. Okay. The probability of these non-enzymatic single electron reductions of oxygen increases sharply under conditions that overfill the coenzyme Q pool as a consequence of increased supply or reduced demand. Okay, so this is saying that you can trace the overflow of electrons by what is it? What is this saying? This is pretty hairy. I apologize. Again, like metabolism is outside my comfort zone, so I have to be really careful here. Yeah, electrons reach oxygen, and then you get overflow when there's too much demand. Then instead of oxygen, you get some leak to the superoxides. Okay, I knew that before. That's not new, right? There's nothing else. The probability that these single reductions of oxygen. Okay, yeah, okay. So the presence of superoxide can indicate that the mitochondria is overwhelmed, too many electrons, not enough oxygen to absorb them. Yeah. They're also cramming a lot of information into such a small paragraph. I actually think that if you're a metabolism guy, that this is probably not that hard for you. But for me, I'm like, okay, coenzyme Q, NADPH. It takes a while to like rack these from my memory. The mitochondria of this particular region are prone to this mode of operation during waking when caloric intake is high, but neurons' electrical activity is reduced, leaving their ATP reserves full. Okay. The mitochondria of the sleep neurons are prone to this mode. What mode? What mode? This is just saying how there's... Okay, maybe this is saying... This is a writing thing. Maybe this is saying that the mitochondria of the sleep neurons are prone to electron overflow. because caloric intake is high, but activity is reduced, leaving their ATP reserves full. So this is saying that when you wake up, you have a lot of energy and therefore not a lot of demand on the ATP synthase. So that's this thing right here. So either you have too much electrons and not enough oxygen to take up those electrons, or your pump, the ATP synthase, stops being needed. And then that produces a backflow. Like if the ATP synthase is not chugging along and smashing those electrons into oxygen, Clockwork, you can correct me if I'm right on that. Then there is an overflow, right? There is a reduced need. The proton pump stops needing to be operated, and then there's like a backlog of stuff. So too many electrons can come from either too much demand upstream or not enough demand downstream, if that kind of makes sense. Very confusing, and I hope I got that right. If I run into something that contradicts that understanding, I'll go back and try to reinterpret that, or somebody in chat can help, because metabolism is kind of like hairy. But it's saying that when you wake, your ATP reserves are full, so the machinery stops being needed to. So you don't necessarily have that electron sink anymore, and then those extra electrons just smash into oxygen all willy-nilly, creating superoxides, which can be kind of bad. Is what I believe this is saying. Indeed, measurements with the genetically encoded ATP sensors showed approximately 1.2 higher ATP concentrations in this region, but not projection neurons after a night of sleep deprivation than at rest. Does that make sense to me? Are more prone to this operation during waking when caloric intake is high, but when neuron is low. And they got a lot of energy to spend. Caloric intake, I believe, is like metabolic intake. Like you're gearing up in your circadian rhythm to tackle the day. Measurements with ATP sensors show that you have more ATP when you are sleep deprived than at rest. ATP concentrations rose acutely when these neurons were inhibited by arousing heat stimulus, which releases dopamine onto their dendrites, and fell below baseline when they were stimulated, mimicking sleep. Okay, so when these neurons are active, you sleep. When these neurons are inactive, you don't sleep. So when these neurons are chilling, okay, I need to like write this down. Hold on. I need to put some text on the screen because this is pretty confusing. Okay, so keeping track. Okay, so like a backup battery. So let me get this straight because I think this is really important for where they want to go. Oh, I'm so lost in the sauce right now. Feels good. Feels good to use my brain. This is the most I've had to think in a while. So DFBN active. This causes you to sleep. DFBN inactive. This is sleep deprived. Waking up is in a way sleep deprived, which is inactivating DFBN, which is causing ATP reserves to be full. Okay, I think that makes sense. I think that makes sense. Oh, you cannot see that at all. That's okay. I figured it out. Let's see. Okay, so I get it. Gosh, this is difficult. This is difficult to explain, but I'm gonna try my best. So these neurons, the DFBNs, these are active when you are sleeping and inactive when you are awake. So when you wake up, these neurons get shut off, and when they get shut off, they don't need their ATP anymore. So their ATP reserves get full. And when their ATP reserves get full, they might be experiencing some electron leak. So I think that is what that means. if this really matters in the future, I will explain it one more time, and maybe I'll draw something quick on PowerPoint or something. But I think I understand it, and I hope I understand it correctly. Metabolism is so hairy. But okay, I think that's what this section is trying to get at here. Okay. Great. So I think I understand that. I think it's also cool for you to see how I try to tackle things I don't understand and figure out things on the spot. I think there's maybe something satisfying, even though for me it's kind of terrifying, but I hope you find it kind of interesting to watch me work things out. Even if the chance of an individual electron spilling from the coenzyme Q pool is low. So I should explain this too. So as commenters have said, the electron transport chain within the mitochondria is the carousel for electrons to form ATP. Electrons need to be passed from different protein complexes to eventually be linked to the final step, which combines ADP with ATP in the ATP synthase enzyme. So coenzyme Q is one of these electron carriers that helps the electron get to its final destination. So if the chance of an individual electron spilling from this electron carrier, right? So if things are running smoothly, then coenzyme Q will pass this electron to the next carrier. However, when things are too crazy in this factory, sometimes you get leakage. So this electron might pass through coenzyme Q, but then it spills out of coenzyme Q and smashes into an oxygen prematurely. However, metabolically active cells, such as neurons, will, by the sheer number of electrons passing through the respiratory chain, generate significant amount of reactive oxygen species. That's fun. I did not know that. I imagine that to be true, but it feels good to actually read that. Your neurons are very energy intensive. Somebody can pull up the stats on like how many calories your brain actually uses per day, but this makes a lot of sense that your neurons are very, very well adapted to I shouldn't say that. Your neurons are using your mitochondria like crazy, essentially. So it makes sense that the mitochondria, the scavenging that is supposed to protect the neuron, is maybe not always active. And so neurons are reservoirs of these highly reactive oxygen species because there are too many electrons because the demand for energy is so high in the neuron. Their anti-cyclical relationship with energy availability, which peaks during waking and energy consumption, which in a sleep-active neuron peaks during sleep, may thus predispose these neurons to an exaggerated form of the electron leak experienced by many neurons in the awake state, making them an effective early warning system against widespread damage. Okay, because polyunsaturated membrane lipids are especially at risk, these neurons estimate the size of mitochondrial leak indirectly by counting reduction of lipid peroxidase-derived carbonyls at hyperkinetics active site. Oh, how is everybody doing in chat, by the way? Because I think this paper, I don't think this is the hardest paper I've ever read on stream, but this is definitely pretty hairy. This is a pretty hairy kind of paper with a lot of metabolic things. But what this is saying is that we can use this hyperkinetic subunit of this linked protein between neuron activity and the mitochondria by determining its electron overflow. Yeah. My head hurts and I'm used to reading neuro papers. Yeah, this is like why a lot of nice innovation happens at the intersection of two disciplines, right? This is not only a neuro paper, but is also a metabolism paper. The words flow into one ear, not the other. I'm hoping to get back into some more cell stuff versus some biochem stuff soon. I'm almost at comprehension, but at that point is where the uncanny valley, and I feel like I understand less than nothing. That's fair. I'm lost, but that's fine, to be honest. I think that's okay. I think I'll try to pull it together when I figure it out myself, because I'm trying to understand it piece by piece. But once things start falling together, I will summarize it for you. Feels good understanding a little, even though I've started learning metabolism. That's good. That's really good, honestly. Like, if you are catching on to the application of what you're learning in class, that's really the best case scenario. Okay, but this is saying that these sleep neurons can act as sentinels for metabolic stress because they get stressed pretty easily and quickly because they are synced to get hit with a lot of energy demand. Or not energy demand, but are quick to shut off, thereby causing everything else to get kind of messed up. Okay, Several lines of evidence point to a mismatch between the number of electrons entering the mitochondrial transport chain and the number needed to fuel ATP production as a root cause of sleep. Okay, so a mismatch between the electrons entering and the demand. So, so maybe it helps to think of this like econ, right? So there's supply. So the supply is electrons, and then there's demand, which is how much ATP the cell needs in the individual moment. So when there is maybe too much supply for the demand, or when there is too little supply for the demand, maybe that's where this is heading. And we'll see which of those cases it is. Oh, I love that analogy, if that's right. Too much demand. I think the title is that, right? A mitochondrial surplus induces sleep. Okay, so this is saying when there is too much electron supply and not enough demand, that is when sleep is induced. And so this is saying that when you are awake, your demand is at an all-time low. Like when you are sleep deprived, that maybe there's too much electrons because your demand for ATP is at its lowest. Okay. Sure. I understand that from a theoretical perspective. Your ATP, oh, that's a great way to put it, Clockwork. Your ATP tank is full, but the metabolic processes are shoving down electrons down the chain. Yes. So there's like an overflow. There's too much supply and not enough demand. And that leak is what's causing sleep. Okay. All of this is like this entire thing is basically building up to this model. And now I believe they are going to test that model, or I sure hope they test that model, because I'm willing to accept this model, but I would like to see some evidence for that. So what are we getting into here? And I want to see experimental causative experiments and not just a correlation. So we'll see if they pull through. Okay. First, opening an exit route for surplus electrons from the coenzyme Q pool by equipping the mitochondria of these neurons with alternate oxidase, AOX, of a different species, which produces water in a controlled 4-electron reduction of O2, not only relieved the basal pressure to sleep, but also remedied the excessive sleep need for flies whose ability to remove breakdown products of peroxidized lipids was impaired. Okay. So here's how they are going to. So they're not experimentally pumping more electrons through. They're actually doing the opposite. They're going to relieve the water pressure on the system. Like, because the ultimate demand, right? The ATP synthase at the end of this cycle, that demand is not changing. But they are going to relieve the pressure valve by providing the electrons a different way to escape safely. So these electrons, instead of being shoved down into this narrow passageway only to leak through coenzyme Q, instead of leaking coenzyme Q because it's overwhelmed with electrons, it's going to provide them a safe place to dump those electrons onto O2, which produces water. And then while doing so, the flies didn't need to sleep as much. This is background evidence, right? So this is a different paper that they're citing. That's not evidence here. Okay, that's how much do I like that? Only on this type of neuron or for every neuron? Just for this type of neuron, the sleep neurons. So let me write this out in the chat. So maybe this will help people who are struggling with this idea, and having it as text, and I'll pin this text, maybe that'll help. Sleep may be the result of low ATP. Okay, let me do it this way. Electrons flow into ATP synthase to make ATP. Sleep deprivation reduces the need for ATP, but does not change the amount of electrons. It does not decrease the number of electrons that get pumped through the mitochondria. Uh, thus, there are too many electrons per too many electron supply and not enough ATP demand. Too many electrons overwhelm the electron transport carrier coenzyme Q. These electrons leak and do something we don't know, damage, and causes sleep, causes the drive to sleep. 6. When you help coenzyme Q with the extra electrons, Drosophila no longer or have a reduced requirement to sleep. Okay, chat. How's that? Does that make sense? This is a lot of complex ideas to mishmash around in your head, but especially if you haven't thought about metabolism in a long time. But I think that's a pretty good way to understand it. And of course, anybody can help check me on that. But that's the model that we're working with. When you are EPI, you have a reduced metabolic demand for ATP in these neurons. Like, don't think about this as body wide. Who knows what the body is doing? But these sleep neurons, when you are sleepy, they kind of don't need as much ATP anymore. But that lack of ATP demand causes a buildup of electrons that leak out and then force the sleepiness to happen. Yeah, I think so. Got it. I see. That's good. I'm happy with that. And I'm glowing because, like, you know, it's kind of like playing Deltarune or Undertale, where there's this boss that you throw your body at repeatedly, and then when you finally beat that boss, there's a feeling of relief and accomplishment. Speaking of which, Clockwork, how's that Sans fight going? Is this in vivo or in vitro? It's a good question. This paper doesn't say, but I'm guessing this is in vivo, this particular citation. So this is not, so this paper is doing it in vivo, but this citation of this metabolic exit routing is from a paper that we're not reading today. But also remedy the excess sleep need of flies whose ability to remove breakdown products of peroxide lipid was impaired. Yeah. Wouldn't the decrease in ATP demand also be expressed in lower glycolysis and therefore lower NADPH production and less electrons? It's a good idea. Will not. I'm not a metabolism expert, but glycolysis, even though glycolysis feeds into the mitochondria, glycolysis also feeds into a bunch of other stuff. It's a teaser, a little bit of a teaser for my next video. I'm not going to tell you what it's about, but what you should know is that NADPH is not only something that can be used at the mitochondria for the purposes of making energy, but can also be used to make superoxides, which are really important for immune cells to be able to destroy bacteria, right? So glycolysis feeds into mitochondria, feeds into the electron transport chain, but it also can feed into other things as well. Okay, let us continue. Second, increasing the demand of these sleep neurons for electrons by overexpressing the uncoupling proteins UCP4A or UCP4C which short circuit the proton electrochemical gradient across the inner mitochondrial membrane, decreased sleep. Okay, so that's so this is increasing the demand of electrons, which short circuits. That's so cool, right? That's awesome. Short circuits the proton electrochemical gradient across the inner mitochondrial membrane. So this is essentially a similar manipulation, but at a different point in the mitochondrial story. Is it weird that I'm feeling bad for fruit flies? No. I mean, I don't feel good about animal experimentation, but I do acknowledge that it is really important to understand basic biology. And third, powering ATP synthase with photons. Whoa! Hold the phone. That's crazy. Third, powering ATP synthesis with photons rather than electrons by illuminating a mitochondrial targeted version of the light driven archaeal proton pump delta rhodopsin made NADH derived electrons in the sleep neurons redundant and precipitated sleep. Whoa! Whoa, you can do that? You can power a proton pump with light? What? What? No way, dude. What? So this is what I was saying with like, I'm okay with going into this as long as the modifications that they make in their experimental system are surgically targeted. So, whoa. Okay. Okay. I need a moment. Whoa, man. Whoa. Clockwork, where are you? You gotta do a new video on a new ATP synthesis enzyme. One that is powered with light instead of electrons coming from the archaea. What the heck? Whoa. I'm freaking out, dude. Whoa, that's crazy. Okay. Let's look at the figures then. Wow, I'm like, yeah, I just got chills. I just got chills. That's crazy. That's insane. This has nothing to do with light affecting circadian rhythm, does it? I believe that's a good question. We're gonna have to see how they pull this off. Because I think this is probably putting a little light bulb in the Drosophila's head instead of their eyes, right? So signals transferring from the eyes into the brain, that might affect it in a more natural way, but this is direct stimulation of neurons, I believe. Hey, fruit flies were used to study gut bacteria and cravings. They're used to study everything. Drosophila are truly, truly incredible. And I think we're about to learn that this paper is really going to show off some of what you can do in a Drosophila. Okay, so we just read the text. Now we get to look at the actual data. Early beta of photosynthesis? Maybe. I don't really know. Nep could maybe say, because I don't have photosynthesis memorized. Smashing things to make sugar is not necessarily the same as smashing things to make ATP. Okay. A mitochondrial electron surplus induces sleep. So this is why I think this is so incredible, because if you keep everything in the mitochondria the same and you don't affect its ability to make ATP, and you just change the ATP synthase, then we can start to say that, okay, maybe I don't agree that it's the mitochondria because I think that's not specific enough, but at the very least, the electron transport chain is what could be driving sleep. Now, that's my idea so far, right? Because when you replace the electron dependent proton pump with one that is photon dependent, apparently that reduces the need for sleep. So at the very minimum, it's the electron transport chain. Whether or not I can say that it's the mitochondria as a whole, that's a little different, right? But oh man, that is very tempting. Does it relate when neurons hardly get light? We're about to see a genetic way to do that, because we are putting an archaea protein into the mitochondria, which will be an artificial system for sure, but one that we can manually turn on. I think that's what this is getting at. So we'll see. A mitochondrial electron surplus induces sleep. So this is our diagram of the electron. So this is their schematic. So let's take some time to dig into this, and maybe we'll zoom in for this. Wow, a proton pump driven by light is bonkers. Okay, so this is our hyperpolarization dopamine. So this is the plasma membrane, and this is the neuron, right? So this is an image of the neuron, and we have this system of pumps that can depolarize the membrane, affect firing. I'm guessing this is what the link is. And then we have our modified mitochondria here. So let's see how this works. Let's first read the caption. Proton pumping complexes 1, 3, and 4 convert the energy of electron transfers from NADH to O2 through intermediates coenzyme Q and cytochrome C into a proton electrical gradient, delta P, across the inner mitochondrial membrane. So this is your basic mitochondrial biology. You use the power of electrons to power a pump, and that makes you some ATP. UCP4 discharges. So this is a protein that you can add in to the mitochondria that can sabotage this system, whereas illumination of MitoDR charges the inner membrane. So this over here, if you power this with electrons, you get a photon gradient, and then UCP4 reduces that, right? So you want to pump hydrogen out and then let it flow. Okay, yeah, so I wasn't prepared to do this, but let me give you a quick 101 of mitochondria, right? So the regular way your mitochondria make ATP is it grabs the high energy electron carriers, and that is going to pump, right? So here we have like electrons carrying these electrons throughout 1, 3, and 4, and this electron transport is going to pump hydrogen outside of the mitochondria. Now, when there is an excess amount of hydrogen on the outside, these hydrogens are going to want to come back into the mitochondria and maintain homeostasis. Now, the wonderful enzyme ATP synthase uses this hydrogen pump to power a turbine that smashes phosphate into ADP to create ATP, which is your cellular fuel. So that's pretty incredible. And these experimental manipulations that they're introducing here is if you want to sabotage this experience, then you can add this UCP4, which enables hydrogen peroxide, hydrogen atoms to flow back into the mitochondria, which is kind of preventing these electron transport carriers to do their job. And if you want to manipulate them in the opposite way, you can introduce MitoDR, which when you smash, this is HV, this is a photon, right? This is a photon. When you illuminate it, that will pump hydrogen peroxide on the outside and then allow the regular machinery to work again. So this is a different experiment than the one that they cited with the... no, this is the same one. Sorry, I think I'm... Sorry, they're not replacing the ATP synthesis. They're not replacing ATP synthesis. Sorry. Okay, I understand now. I was a little incorrect before. So the regular way things work is that electrons flow down this electron transport chain. You pump out these hydrogens. Those hydrogens flow back into this enzyme. You get ATP. Now, if you want to remove the electron transport chain from the equation entirely, what you can do is you can instead force the mitochondria of the Drosophila to make this enzyme, which pumps, it does the same thing as the electron transport chain, right? It pumps hydrogen out, but instead of requiring an electron to do it, it needs a photon. So you can shine a light directly onto the mitochondria, or in this case, the cell. And as long as you have that light turned on, it'll be pumping that hydrogen out. So an incredible manipulation here. So essentially, we can ditch the electron transport chain and we can see if it is energy or is it the electron transport chain. So remember at the beginning of the stream when I was like, how are they going to sabotage the electron transport chain without affecting ATP synthesis? And this is their answer. They are going to sidestep the electron transport chain by giving the Drosophila mitochondria a pump from archaea, which are crazy. Absolutely crazy. Hey, Nep, welcome. Nep being my fellow V-Theory member who is a horticulturalist. We were actually just talking about photosynthesis earlier on in stream. Are all turbines derived from cilial motor development? Like, is there convergent evolution there, or is it from a singular source? I actually don't know. That's an interesting question. Okay, so here's what they're trying to do here. If we sidestep the electron transport chain, we sabotage it using this protein, right? This protein, is that what this protein does? Let's continue to read the caption. The return of extruded protons to the matrix spins the blades of the ATP synthase. I love that, right? Even they call it a turbine, and produces ATP, which leaves the matrix via CESP in exchange for cytoplasmic ADP. Neuronal ATP consumption is activity dependent in part because the plasma membrane must restore ion gradients dissipated by action and excitatory synaptic currents. So this is why the neuron has such high energy demand, right? As soon as you make that ATP, these pumps that maintain sodium and potassium balance across the membrane are immediately using that high level of ATP to make sure your neurons can actually fire. We have to look into Rubisco someday. We absolutely should. I'm scared though, because it's been a long time since I thought about plants. An oversupply relative to ATP demand of electrons to coenzyme Q increases the risk of single electron reduction of O2 to O2 minus at complexes 1 and 3. So this is saying again that when there are too many electrons, you can get leakage and you can produce O2 minus, which is bad and toxic. Okay. That is what this is saying. So how are they decoupling things, right? Because, yeah, I can't understand when neurons can sense light, or does it signal from eye photoreceptor neurons? What is the name of this neuron? I don't know what the name of the neurons are, but there are photoreceptors in your neurons of the eye, which can communicate to the brain as a whole whether or not you're experiencing light. In this situation, I think they're just shining a light through the Drosophila's head. It's a lot easier than that. The photon will smash into this enzyme and then that'll pump protons out. I need to reread exactly how they're doing this. Increasing the demand with short circuit the proton electrochemical gradient, decrease sleep, powering ATP synthesis with protons rather than electrons. Okay, I think I get it. I will continue to go and look at the data. What is the relationship to the TIM/PER mechanism that is already known? Per-TIM buildup during the day is thought to promote wake, but has a negative feedback mechanism to stop its own transcription. That's a good question, Random Trash. I'm not a sleep expert, so I actually don't know. Maybe if we go through the paper, you'll be able to offer some insight, because I'm unaware of how sleep is regulated, especially in Drosophila. Okay, so let's look at the data here. Summed intensity projections of the sleep neuron dendrites expressing whatever this is, plus RFP. So what is this? Where did you define this? This is the ATP sensor. Okay, so expressing ATP sensor and red fluorescent protein plus something or another, according to the keys below, increase after sleep deprivation. Okay, so this is RFP. This is, oh, let me make sure that you can see this. There we go. So let's look at the data. All of this reading, and we finally get to look at some actual images. So these are the sleep neurons, and the RFP, red fluorescent protein, signal is going to correlate with the amount of ATP in these systems. So chat, we finally get to interpret some data. We finally get to get in on some of this. The mean green-red ratio. So green, I believe this is just their general sleep neuron stain. So green is going to give you the location of the neuron. And then the red is going to tell you how much ATP is being made. So on the left here, we have the Drosophila that have slept normally. And then on the right, we have the Drosophila that have been sleep deprived. So chat, what can you tell me about the ATP demand in, not ATP demand, sorry, the ATP supply in sleep deprived Drosophila? I like this sweaty distressed fight today. I'm sweaty and distressed because I had to replace my microphone stand with my phone stand because I broke my microphone stand. Why is it monochrome? I don't know why they did it monochrome. They converted the green-red ratio to a monochrome scale, which I think is a little sad. So you can imagine that the real picture is green with red on top, which is hard to see for colorblind people. And then when you calculate the ratio between green and red, you can convert that into a pixel value. So that is why it is monochrome here. Give us the color. If they did green, they could have false colored it for sure. I'd like to see pretty pictures, so I'm personally disappointed that they're not that colorful, but you know, whatever. So yeah, chat, what can you tell me about the energy supply of sleep deprived neurons? And I think when you look at the data yourself, you might be able to put together some of these pieces for yourself. Remember that the left panel, these are flies that have slept normally, and then on the right, we have sleep deprived Drosophila. So sleep deprived neurons, how much ATP do they have compared to neurons that have been rested? Come on, chat. I know you can do this. It's been a while since I've made chat interpret some data, but I believe that you can do it. It's been a while, and I want to hear your thoughts. None? What do you mean by none, Hero? And this diagram is for this. Like this is one figure, these two combined. So maybe that'll help. I'm going to take another sip of my chamomile tea. That's what I'm drinking today. Sleep deprived neurons would have less ATP, right? Is that what the data is saying? So the green red ratio is indicative of the ATP sensor. So what does it look like here? It's a little complicated for sure. But yeah, so Guy says sleep deprived neurons have accumulated ATP during the awake state. And yeah, so that is what I am seeing as well, right? So that, yeah, exactly. They would have more in reserve, right? So the brightness here, the green red ratio, the brighter this picture is, the more ATP these neurons have. So when you are sleep deprived, your sleep neurons, again, don't think of this as general body physiology, but specifically your sleep neurons have way too much ATP in the bank, right? They have banked up way too much ATP. And this is possibly because there's not enough demand, there's not enough flow through this system to deal with this, right? So this is correlative, right? This is saying we have this relationship, but this isn't indicative of a causative relationship between mitochondria and sleep yet. This is establishing the given context. So they aren't using as much ATP as they are making it. Exactly. They're letting their ATP build up when you have sleep deprivation. I got the demand and supply mixed up. I did too multiple times while reading this paper. It's not an easy paper to read. Okay, what about figure C? So let's read the caption for figure C. Whoa! Figure C. In rested and sleep deprived plus RFP or A team. What's A team? What is A team? Okay, so it's another ATP sensor. So they're using two different ATP sensors, and yeah, I'm not going to go over that. They find the exact same relationship, right? When you have sleep deprived sleep neurons, those sleep neurons have too much ATP compared to the rested neurons. Sorry, just changing the zoom on the page for chat. A-team theme starts playing. Okay. Arousing heat elevates ATP in these neurons expressing the ATP sensor plus tomato. Why? Okay, so now we're using heat, and heat can be a way to control some genetic circuits, and I'm wondering if we are seeing some of those genetic circuits, because I'm not really understanding. Fig.2D. That's annoying. Fig.2. Fig.2A, Fig.2B, Fig.2A, 2F, 2A, 2G, 2A, 2F. Okay, so there's no. That's annoying. So what is why are we heating up? Huh? Why are we heating up stuff here? Heat. This is not what I'm looking for. Ephys, heat map, heat stimulus. Okay, I see. Okay, so they are heating up the neuron. I don't know how though. Are they just baking them? Okay, I don't really understand that, but okay. So apparently, if you heat up these neurons, that will inhibit the action of these sleep neurons. So it'll prevent them from working. So it is going to mimic sleep deprivation, right? Because these sleep neurons, when they are active, you are sleeping, right? These active neurons will actively make you sleep. So if you break them by heating them up, they will mimic sleep deprivation. So in this figure, we'll see if that is true. So let's go ahead and zoom in once more. So here we have time on this axis, and this is delta r slash r. What is r? Rest and relax? Like, what is this? Figure D. Um, change in fluorescence intensity ratio of the ATP sensor. Okay, so this is the delta R over R. So the fluorescence intensity of what? I'm a little confused. I'm a little confused with co-expressed D tomato relative to pre-stimulation baseline. What? What? Plus D tomato fluorescence. Oh, okay. This is the ratio of the ATP sensor to the neuron marker, which is DT tomato. So this is the difference between This is the same thing as this up here. It's just shown differently because we're on a time course here. So instead of just measuring light, we're measuring the ratio of that light over time, which is why we don't have a pretty picture, but we have a line graph. Yeah, it's the intensity of the light, but it's comparison with the light of the neuron itself. So when you apply the heat, you get a spike in the levels of ATP, and it looks like it stays that way even after when you remove the heat, which is interesting. So you manually, with the power of heat, quote unquote, sleep deprive these neurons, and then you get more ATP. That's kind of cool. Okay, and then E, A, B, C, D. Where is E? Optogenetic stimulation dissipates ATP in the sleep neurons expressing the sensor and crimson, but not in the sleep lacking trimson? Okay, what is this? What is all of this stuff? Flies expressing gate open the conductance. Doesn't explain what that is. Doesn't explain what that is. Okay, I'm gonna Google what this is real quick. CS Trimson is a light-gated cation channel that can be used as an optogenetic tool to activate neurons. Okay, so this is a way to manually activate, but activate in what way? Hold on. It is optimally excited by light, an artificial channel rhodopsin. Okay, functional channel link, covalently linked all transretinal chromophore. But what are you linking this to? I don't really understand what this crimson is linked to. Optogenic stimulus. Oh, okay. No, I don't get it. Hold on, I don't get it. I don't understand. Optogenetic stimulation dissipates ATP, but Why? I'm struggling here, chat. I am struggling. um, the summary of where we're at so far is that when you sleep deprive your sleep neurons, they they get a backlog of ATP. They get too much ATP. Now I'm trying to figure out what this figure is about. Um, I'm not having any luck. So this is the neuron that is these are the sleep neurons. They are expressing the ATP sensor, and then when you give them this optogenetic channel, but it's not linked to anything. What is it linked to? Hmm. Like I don't know what this is. So optogenetics, right? This is a tool that you can use. You can shine a light on it, and then it does something. But I don't see what this is supposed to do. Why would more ATP make you sleepy? Seed. It is not that it's more ATP that makes you sleepy, but it's a symptom of what makes you sleepy. What makes you sleepy is too many electrons. Because if you have too many electrons, it could be an indication that you have a buildup of ATP. You can kind of think of it like the flow of water, right, down different uh, ponds, I guess you could say. And when that last pond is full, you start to get a buildup of water in the back, in the further up in your plumbing, right? So it's not the ATP that is the issue. ATP is just the sign that there is buildup in your plumbing. I think that's the best way I can describe it. I'm really bothered by whatever this is. Let's figure 2e, figure dot 2. What is this? What is this actual channel? I don't actually understand what you are optogenetically modifying. Okay. ATP concentrations rose acutely when these were inhibited by arousing heat, which releases dopamine, and fell below when they were stimulated, mimicking sleep. Okay. Yeah, I mean, it helps to just say what this is supposed to be. So it's a cation channel, but I think what's important to say here is that the cation that this thing lets in is the cation that is required for neuron firing. So let me break that down for chat. So let's take a look at this figure. Let's zoom in. You got this, cell boy. I totally got it. I understand now. So in this gray line, we have our sleep neurons that are expressing the ATP sensor. And when you shine a light on the neurons, nothing really happens in these neurons, right? Because these are just the normal neurons. There's nothing fancy about them. They're just your sleep neurons, and they have a fancy ATP sensor. And when you shine a light towards them, you can see here that they don't respond, right? But when you put this channel, this CS trimsin, into the cell, this is a optogenetically responsive channel, meaning that if you attack it with a certain wavelength of light, the channel opens and allows the influx of cations. In our case, these are the cations that are required for neuron firing, so probably calcium is what I'm guessing, right? So in the orange line, we have neurons that can be programmed to fire when you shine a light on them. Does that understand? This is actually really important because we're going to talk about optogenetics at some point again later on in this in like my channel. So I want to make sure that I practice how to explain this. Gray, we have our normal sleep neurons that are just chilling. And then when you take your sleep neurons, you give it a channel that responds to light and lets in calcium to start neuron firing. That's what's in orange. So when you have a system where the channel can open when you attack it with a certain wavelength of light, you can essentially manually turn the neuron on. So what this is saying is that when you manually turn on your sleep neurons, the ATP level crashes. Does that make sense? See, the poll is to see if my explanation for optogenetics makes sense. You can program a neuron, you can force it to fire if you genetically modify it to express this protein that responds to light. So in this 2-minute block, this is the block right here. This block right here, this is when the light was turned on. So when the light was turned on, the sleep neurons are firing like crazy. And you can see when they start firing, the ATP levels crash. This is perhaps not surprising, right? Because we know that when neurons fire, they need a lot of ATP. But this is showing the reverse relationship, right? When your neurons are inhibited, but when the sleep neurons are inhibited, right? Like when in sleep deprivation, then you get an increase in ATP. But when your sleep neurons are active, then you get a decrease in ATP. Okay. Very, very cool. Still, this is all just consistent with their hypothesis. They haven't actually tried to break anything quite yet. Okay, so we move on to these. Ooh, okay, sleep per day in terms of hours, and then what is this? Okay. Okay. Let's read the caption here, because I think this is pretty interesting. There's no way that I'm gonna make it through this entire paper in this amount of detail, but I figured this first figure is probably the most important one for establishing the relationship. I'm just taking a look to see how much. Oh, actually, there are only 4 figures here. Maybe we will finish this today. Yeah, because we're halfway through. Okay, so F, G, and H. What are you? Sleep and fly is expressing the marker for the sleep neurons, and V-glut gal4 driven. Okay, so V-glut gal4, I'm guessing this is a pan excitatory neuron driver. So this is only expressing these sabotage proteins and parental controls. Sleep, okay. V-glut, right? I think so. So then maybe in sleep, the ATP loaded neurons are fired in a way that cleared it up. Exactly, yeah. That's what they're trying to mimic, because instead of actually letting the Drosophila sleep, they are testing it more directly by just forcing the sleep neurons to fire. So you can make the relationship with how it happens naturally. So this is like surgically applying the process of sleep without requiring the whole brain, right? Just requiring just the sleep neurons itself. How much do I want to get into here? What is this V-glut system? Maybe I think I'm maybe happy with this so far. I might just skip these because maybe I'll do this one, this middle one. I don't know what F. It's kind of important. I think I'll figure it out. I'll figure it out. So why are we using V-glut? What is this? Parental controls. Driven without the need. Because I don't think I don't really know. Okay, so let's zoom in here. I'm having a little trouble interpreting this. I could probably figure it out given enough time, but I kind of don't want to spend the time on this figure, but I'll try my best here. So this is a way that you can denote a complex combination of different experimental conditions. So this one has the V-glut marker. This is our control. This is just our regular neuron. I don't know what this upside down U is, which is kind of interesting. And in this category, the only thing that is active is this marker. And I think this is just like a naked promoter, I think. So this is right here, our control. So the Drosophila are regularly sleeping, I don't know, like 16 hours a day or something. And then when you express this thing that sabotages the electron transport chain, but you don't put it in the sleep only neurons, you get no change. And in this one, you get the sabotage protein, the one that pumps that allows protons to flow back in and thus ruining the electron transport chain. When you put that into the sleep neurons, you get a decrease in sleep. So does that make sense? You don't sleep as much. You sabotage this. So you allow this to happen. But why does that work? Why does that work? I am confused. Time to read the paper. Can't figure it out from the caption, so I must read the paper. So this is F. Big 2 dots. Figure big dot 2. Big dot 2 F. Okay, short circuits and decrease sleep. Short-circuited the proton electrochemical gradient across the inner mitochondrial membrane and decrease sleep. Activating UCP4 would inhibit ATP production, right? That is true. Because by pumping the protons back into the mitochondria, you're ruining this gradient. Yeah, okay. Yeah. The electron transport chain is trying its best to pump stuff out, and by ruining that cycle, you are ruining the efforts by the electron transport chain. So you have too much hydrogen on the inside to pump out. So there's, I believe, less chance for electron leak because you have more hydrogens inside. Yeah. Okay, that makes sense. So by expressing these sabotage proteins, you sabotage the electron transport chain, which decreases the chance that you have electron overflow, which causes you to sleep less, is what the correlation is, I believe. And then if we have the opposite, if we increase the amount of ATP by giving the mitochondria proton pump, then what happens? Very, very tricky. Third, powering ATP synthesis with photons rather than electrons. Precipitated sleep. Right. Okay. So if you give the mitochondria another way to pump those hydrogens out, you actually see that they sleep more per day. Oh my God, 30 hours of... Oh, minutes per hour. Okay, never mind. It's a little bit of a different metric than the previous experiment. Okay, so both of these experiments are saying that if we increase ATP, we increase sleep in the sleep neurons. And then the vice versa is true. The thing is that the regular ATP machinery is still intact. So I don't really know if I buy that mitochondria are powering sleep versus just ATP supply. Because you gave it more ATP by giving it this, which is correlating with the action of the electron transport pump. Right? Because ideally you would want to shut this off, maybe with actual cyanide. Like if you kill this with cyanide and then relieve the pressure with this, maybe you could say that it's not electron dependent. Yeah, that's kind of confusing to me. That's tough. So here's the reason why I'm confused, chat. They powered the proton rather than electrons. I don't know if this is rather than, because I don't see any indication that the regular electron transport chain isn't still active. So that's a little suspicious to me, because this is still functioning, presumably. H. After cumulative sleep percentage, photo generation, pairwise comparison, sure. A, B, C, D, G. What is retinal? With or without retinal and parental controls? What is a retinal control? Oh. So a retinal control is what? To prevent the undesired activation of optogenetic actuators or the photoconversion of all transretinal by ambient light, flies expressing the crimson or mitochondria and their controls are reared and housed in constant darkness and transferred to food supplemented with 2 all transretinal. But I don't know what that is. What is this thing? 2 millimolar all transretinal molecula. What is this reagent? What does this do? Is a retinal in which all 4 double bonds have trans geometry. Well, what does this do? Chromophores that bind to opsins. Photosynthesizer. Okay. So, if I'm understanding this correctly, this is the full system where you are using that archaea pump. This archaea pump, when you don't add this food or whatever, this food helps with turning on this pump. I don't know what the food does, but I think that's what this is. So when you add the food, you add the archaeal pump that is activated by light. In the sleep neurons, you get more sleep, presumably because you are providing more ATP, or you are causing more electrons to be floating around. Right, because it's hard to decouple this in your brain, but what this paper is saying is that it's not necessarily the ATP that is the issue, it is the excess electrons. So... But, okay, but I'm failing to see how activating this pump causes more, because they didn't shut this part down, right? They didn't poison this. If they removed this and only had this, that would be better, I think. How are you doing with this mechanism? I'm very confused. Clockwork, if you want to go to figure 2G, they are using a archaea proton pump that, oh, this is a great time for you to come back, by the way, because, yeah, in order to decouple the electron transport chain from ATP synthesis, they are programming these mitochondria with mitoDR, which is an archaeal pump that converts, instead of electrons to protomotive force, but rather photons. So these photons cause this pump to pump out hydrogen to make sure that the regular production of ATP is still normal. The problem that I'm having is that this experiment is showing that when you give these sleep neurons this photo-powered pump, that they sleep more. But this is not decoupled from the electron transport chain because I fail to see any indication that the regular electron transport chain has also been shut down. So it's totally possible that this regular electron transport chain is still active, in which case I don't know that this says anything other than giving mitochondria more ATP in the sleep neurons makes the Drosophila more sleepy. That could still be energy, even though their argument is that it's electrons. So I don't want to move on because it's like a big deal for the paper. I'm very confused. I'll put in a good effort for like another 2 minutes to try to figure it out. Because this part is fine, right? When you add a channel that causes the mitochondria to leak protons back into the mitochondria, you reduce the amount of ATP. I guess I don't know how intracellular concentrations of protons affects the electron transport chain. Actually, Clockwork, if you are here, that might be an interesting thing to discuss. Like, I don't quite remember what effect the actual presence of hydrogen has on the electron transport chain itself. Because I always thought that the electron transport chain was driven by oxygen, and that the electron transport chain drives protons, not the other way around. Excuse me. Okay, so unfortunately, I think a lot of what I think about this paper is going to hinge on interpreting this particular figure. And that's very difficult. I think it's pretty hard to make heads or tails of what this is supposed to tell us. Because this is, yeah, it says instead. That's what I'm hung up on. Because it's not instead, it's in addition to. And I don't want to rag on the authors if I'm misunderstanding this, but it's like pretty important. Pretty important. Powering ATP synthesis with photons rather than electrons. But you didn't remove the old electrons. 2A. Made NADH-derived electrons here redundant and precipitate sleep. But that's not... Okay, maybe this is a legitimate gripe. This is assuming, this is making an interpretation, right? I gave the mitochondria another way to pump hydrogens out so that we can get more ATP. The interpretation the authors are making here is that this is making the NADH-derived electrons redundant, and that is why they're sleeping. They actually didn't test that. That is why I've been stuck. Oh my god, that's why I've been stuck on this figure for like the last 15 minutes. Because what they want us to believe is that this is a story about electrons. So I was looking for proof that it's electrons. There is no proof that it's electrons here. It is filling the gap for you. And this is why it's so frustrating to read papers like this where they are trying to pill you into their conclusions, right? The text here should say that providing an additional mechanism by which hydrogen ions can flow out of the cell precipitate sleep, right? This stuff about making NADH-derived electrons redundant is an interpretation of the data, but it was not experimentally derived. So this is the first science thing that I think is annoying, right? In principle, theoretically, if you provide the mitochondria an additional route to pump protons out, theoretically you don't need the NADH pathway, but they never proven that, right? How would you do that, chat? I don't know. This is a test to see if any of you have watched Clockwork's video, or testing your understanding of Biochem 101. And it's okay if you don't understand this. I may have mentioned it earlier in stream, but there is a way that you could poison the electron transport chain and make it and turn it off and replace it with this pump. Does anybody know how you can poison the electron transport chain? Very famous poison out there. One of the most popular poisons to include in works of fiction where assassinations are a thing. How can you poison the electron transport chain? Anybody in chat know? I am so lost on this one. It's tough. Cyanide, exactly. So I'll go over it. Clockwork, if you're confused, I'm guessing a lot of people are confused here. And I'm also confused, right? Because their argument is very confusing. Their whole argument is that it's extra electrons that are driving the need for sleep. So how do they try to prove that here? They genetically programmed the Drosophila to have this channel that pumps protons out without the need for electrons. And when they do that, they find that, yeah, the Drosophila sleep more. That's what this figure is showing, this guy right here, right? When you force your sleep neurons to pump protons out without, in addition to the electron transport chain, they get sleepier. However, they never shut this part down, right? This electron transport chain still works, presumably. It is their interpretation. It is the Kool-Aid that they want us to drink that this is redundant. We don't know if this is redundant. We don't even know how good this works. And that's why I think this is kind of frustrating. And yeah, it's cyanide, right? If you include cyanide here, you block the electron transport chain from happening. I think it poisons cytochrome C, but I don't quite remember particularly. But like some sort of experimental manipulation where you shut off the electron transport chain and then replace it with this light-based thing, that would be interesting, because then if you have no electron flow here, you would hypothesize that you would be awake, right? If you have extra electrons floating around, that means you sleep. You get sleepy. If you have no electrons around, that means you're awake. Yeah, I think so. God. That's tough. That's tough paper. In any case, I don't like that they didn't poison the electron transport chain here. They either established that proton flow out of the inner mitochondrial lumen inhibits the ETC, or that dynamic is like super established in this field. I don't think they do. I'm not convinced by the text here that that is the case. Like, they just assume that we understand that these are somehow redundant. But that's not cited. That's the argument that they want to make in their figure, right? But I don't buy it. Maybe they're right. Maybe the NADH carrying electron transport chain is actually redundant in these cells, but they have not shown it with their own data, and that is pretty annoying, I would say. Ooh, okay. Because this finding, right? This finding that if you pump more protons out, then you get sleepier. This is not different than saying the more ATP you have, the sleepier you get, right? The more ATP, or does it? Or does it? You have more ATP that is usable. This is sleep. This is not ATP levels. This is sleep. Does that change my interpretation of things? Wait, are we sure the photon-powered pump doesn't use the electron transport chain? Maybe the photon just replaces NADH. According to them, the photon-powered pump doesn't use the electron. That it uses photons instead. According to them, right? I actually don't know how this thing works. Right? Powering ATP synthesis with photons rather than electrons. Yeah. Yeah, they sleep more. They sleep more, which means they have less ATP, because when they are sleep deprived, they have more ATP. They're sleep deprived, they sleep more. It's a lot to juggle. The more I listen to Phi speak, the more appropriate the sweaty PNG looks. I am sweating up a storm. I don't think I've ever gotten quite as hard stuck in a paper. I think it felt easier than some other papers, but eventually I overcame them. Now I'm like super hard stuck because there's a lot of ideas to juggle all at the same time. Stuck on this one freaking paper. One freaking figure, I mean. Yeah. This figure sucks so bad. Hydrogen being pumped into the lumen by photon pump. Yeah. The photon. Wait. What? Wait. It's hydrogen pumped out of the inner. Oh yeah. Oh God. Between the inner mitochondrial membrane into the outer, in the lumen between the outer mitochondrial membrane. Yeah. Yeah, I think I may have been mixing up my language, which I'm confused by. Thank you for checking me on that. Yeah. Sorry. That might have been my fault. UCP4 pumping protons into the inner mitochondrial membrane, and then mito pumping out so that it can come back into ATP synthase. But I still don't know how this experiment shows anything about the redundancy of the electrons, right? Yeah, this is such a hard, and you can see that like when writing isn't clear, scientists get hard stuck like this and then get really sweaty like I am right now, right? Because this is making me feel stupid. And a good paper shouldn't make you feel stupid, even because like there's this sense of like, oh, the complicated science, it must be such a good paper if I can't understand it. No, it's a good paper if you can understand it, and I'm just not understanding it right now. Proton pump makes electron transport chain redundant, but electron transport chain works no matter what. Phi's issues still stand. Yeah, okay, thank you for providing a second eye on this. It is kind of crazy, right? Because nowhere are they showing that this is redundant. You know, this is too much. They're trying to get you to buy into this too much when they don't have to, right? I think this experiment is very cool. Like the data speak for themselves. We don't need you to pre-digest our food for us, right? We're not babies. We don't need you to masticate our food and then spit into our mouths, right? Which is kind of what this is feeling like. And it is that disgusting that they are trying to do that, in my opinion, while reading this paper. Again, I remember when I was really excited. It can turn back very quickly based on single sentences that make you just question your own sanity. Yeah, so yeah, I don't like the way that they worded this. This figure itself is fine, but I don't like the way that they described it because it's making me crash out, guys. I'm crashing out. Because it's not even a coherent argument, right? What does it mean for the electron transport chain to be made redundant? Are there more electrons out here? Are there less electrons out here? Like, what are we saying? What are we actually saying about the level of electrons when you put this in? Because to me, all this is saying is that you have provided another way for that you are reducing the amount of ATP that can be made. That increasing. So that's confusing. Pumping out so that ATP can be made more. All right. This isn't an electron specific data point. This is just saying when you pump more protons out that you get sleepier. That's not very convincing with regards to their electron story. And if they didn't include that wording in here, I wouldn't have even given it a second thought, right? Because maybe eventually this argument makes sense, but right now in this given data, it's just like, this is just like not what that's not what that's saying. Or I'm going crazy. And I don't know. I don't know. Maybe I'm going crazy. But in any case, it's hairy, I should say. Okay, I think this is probably the most hairy and complicated important paper, and then this rest of the stuff I think should go pretty quick. But I'm actually going to take a quick break just to vacuum up some food because I am starving. So why don't we go ahead and take like a 10-minute break? I will be back in 10 minutes. So I'll be back at my time, noon sharp, and we'll get back into the paper. All right, so be right back. I am really hungry, but I do want to finish this stupid thing. Be right back. Whoops, Mike. Sorry. I'm extremely muted. Thank you so much. I did drink water. Thank you so much. Thanks for hanging around. Hey, Leo. Welcome, welcome. Um, we're back now, though, right? Surely. Surely. Yeah, I wanted to mute my mic to make sure that you didn't hear my microwave go off. Um, yeah, so Clockwork just sent me the source for the paper that introduced the mitochondria as a way to manually pump out protons using light. And it has nothing to do with checking whether or not the electron transport chain has been made redundant. So I think I think I'm still right to be kind of miffed that the authors of this paper seem to be very insistent on putting words in my brain, and I don't like it when that happens, right? Like, I want to be shown the evidence, and then the best part, like, the best part, even for content creation, right? It's to read and look at the data and then form the idea in my own head and make the connection myself. When other people try to make connections for you, it's like pre-digesting it, right? And like that's kind of what I do on YouTube, right? I'm pre-digesting papers for you to enjoy, but like I'm supposed to be reading the raw material, right? I'm supposed to be getting it as fresh and as least processed as physically possible. It's like going to a Whole Foods and instead of buying, whatever organic pine nut, it's like just Snickers or whatever, which are, fine, but you went to Whole Foods to buy expensive organic pine nuts, right? You didn't go for a Snickers bar. And that's kind of like what I'm feeling right now with the insistence that they it's also like it's like an insult, right? Like, why you don't think I'm smart enough to make my own conclusions, so you are going to push your particular narrative as hard as physically possible. Like, I can't come to the same conclusion. Even if I don't, it's not bad. Like, again, I think these experiments are very cool. I'm a big fan of these experiments. I just don't like that they are making it into baby food. And like baby food that isn't even correct. Okay. Let's shake off some salt and get back into reading this. Oh god, we're only halfway through. Okay. Oh boy. Given this, oh god, dude, come on. Dude, shut up. Dude, shut up, dude. Stop. 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 This is, you know what this is? This is what is it? I'm not gonna say the words on stream because you definitely know where I'm about to go with this, but this is self, you know. Thank you, Seed, for joining the Shedder cells. Thank you so much. Given this wealth of evidence, what 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 are we talking about here? Who says that in their own paper? In their own paper, given this wealth of oh my god. Stop. That's insulting. Also, what wealth of evidence, right? Like, this is plain bad writing. Like, are you referring to a certain set of papers? Like, are you referring to this paper as a whole? What is this in this wealth of evidence? Leo says this feels ChatGPT'd. I don't know. I'm not an expert in sussing out ChatGPT. What I can say is that if it was ChatGPT was feeling itself with this one, right? It was very self-serving. This is extremely pat myself on the back. I'm a good boy. Look at me go. This is very unusual for a paper to congratulate itself like this. Given this wealth of it's like worse. I've seen this sometimes before in other papers that are like, there is a wealth of evidence that suggests, but it's just that more condescending when it's given this wealth of evidence. It's like, oh my god. Hey Mylan, welcome. You came at a good time where I'm mauling about this paper. Mylan, is it regular in your field of discipline for papers to pat themselves on the back like this, given this wealth of evidence in the results section? Not in the intro, but while talking about data. Um, that's crazy to me. That's so annoying. The pat yourself on the back was a ChatGPT 4.0 habit, I believe. The models have their own dialects, interestingly enough. Maybe. In the intro, yeah. In the results, no. Yeah, yeah. I think that's the candidate for research hell, right? Like, this is supposed to be an objective piece of description. You should not be interpreting this for us. You know, Neil, when you said ChatGPT, that may be true because in a results section, you're not supposed to be interpreting data. You're not supposed to be assigning confidence subjectively in the results. That is a strictly intro and discussion kind of thing. Maybe it is ChatGPT because I can't imagine researchers being this free to do something like this, which is just, it reads so poorly. It reads so bad. Also, wealth of evidence from a single paper. What? Yeah, I mean, it's not clear if this is referring to previous experiments or whether or not it's referring to experiments here. But like, this attitude has been pretty prevalent in this entire paper. It's embarrassing. Yeah, Mylan, let me read back here for you. Like, the strength of these statements: sleep, like aging, may be an inescapable consequence of aerobic metabolism. It's a lot of strong opinions where I'm not used to seeing strong opinions. Um, Bonkers. Okay, whatever. Well, this is about the science, so let's continue reading about the science. However, it remains ambiguous whether the upregulation of transcripts encoding mitochondrial proteins signals a net increase in mitochondrial mass or a compensatory response to organelle damage. This is the first time that we've heard of mitochondrial mass. Like, it's kind of weird to just bring this up, and then there's no citation here. There's no citation here, so this is not referring to like a general increase in mitochondrial mass. B, if you were going to introduce the idea of mitochondrial mass, where's the link to the figure? What? 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 Did I miss it? Hold on. Did I miss an entire figure where they talked about mitochondrial mass? Nope, that's the first time they bring up mitochondrial mass. Right? Figure 1, nothing to do with mitochondrial mass here. Figure 2, nothing about mitochondrial mass here. Figure 3, number, volume. Yeah, it is in figure 3. The way that this should have been written is that blah, 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 transcripts, so, you know, under sleep deprivation or whatever, mitochondrial mass increases or decreases. This is kind of introducing an idea in the wrong order. Weird. Weird. Wait, maybe it is taken for granted. Electron transport chain powered by Krebs cycle, which is regulated by metabolic concentration. So sufficient ATP equals ETP. Shut down, no need to test. I don't really know if that is true. If it is true, they should have mentioned it. Electron transport chain powered by Krebs cycle, which is regulated by metabolic concentration. It's just so many different. No, I'm going to be pretty bullish on the fact that I want to see evidence that ETC is no longer functioning when you because even if that were true from a theoretical perspective, you should at least cite a paper where that was shown. And the paper that you had linked had nothing to do with first it was looking at apoptosis or something like cell death. It wasn't even looking at like the flux through the electron transport chain. So like it's just a claim that came out of nowhere. Ugh. Who reviewed this paper? I don't know. It's a Nature paper, so presumably somebody did. This is unfortunate. Or maybe it's not that bad. I don't know. If you're the author of this paper and you want to tell me how dumb I am for not liking your paper, I'd be very happy to like chat about it. Like what the logic was with regards to certain sentences or ways of phrase, or bringing up mitochondrial mass as something linked to an additional idea, upregulation of transcripts, even though you didn't bring up mitochondrial mass to begin with. Like anyway. Reviewers typically wouldn't comment on the quality of writing unless it actively hurts the evaluation of the science. I kind of feel that it's messing with my ability to look at this paper politely. uh, Again, the science is cool. I really hate how this paper is written. To disambiguate these scenarios, we labeled the mitochondria of the sleep part of the brain with matrix localized GFP, mito-GFP, and an independent series of experiments imaged the neurons' dendritic fields by confocal scanning laser microscopy or optical photon reassignment microscopy. Don't know what this is, but you know what this is. Both forms of light microscopy were validated against ultrastructure automated morphometry of deconvoluted image stacks, established that although both optical methods overestimated the true size of organelle size to some extent, the size ratios of mitochondria and DFPNs and uniglomerular olfactory projection neurons in the anterior lobes matched that determined by volume electron microscopy. This is of what? We label the mitochondria matrix localized mito-GFP. Okay. I'm once again confused. Oh, mito-DR. Okay, never mind. Not confused anymore. It remains ambiguous if the upregulation of mRNA encoding mitochondrial proteins signals a net increase in mass or a compensatory response to organelle damage. So we saw earlier in the genetic data that when you are sleep deprived, you have a bunch of mRNAs that are upregulated that have to do with your mitochondria. And apparently, again, they don't mention this. They don't mention this mass thing anywhere else. Is the first time we're seeing it, but apparently your mitochondria get bigger. And the reason why they want to determine is this because what? Oh, wait, now I'm confused again. It remains ambiguous whether the upregulation of these mitochondrial transcripts encoding mitochondrial proteins signals a net increase. Okay. So you get sleep deprived, you get more and more. Sorry, I was too mad. This is not the writer's fault. This was my fault for misreading it. Okay, I take it back. I take it back. This sentence doesn't bother me. I just read it wrong. They saw an increase in mitochondrial, in mRNAs that correspond to mitochondrial regulation. And they want to know whether or not that's making mitochondria bigger or repairing them. So that's fine. I read this wrong. But again, I read it wrong, I think in part because I'm mad at other things in this paper, right? When you are tilted, you often can't do a good objective job reading stuff like this. So that makes sense. I'm just mad that I got mad. Okay, so you have a spike in mitochondrial mRNA stuff happening. Is that making, and I don't know, it's just weirdly worded. Signals a net increase in mitochondrial mass. Maybe this is ChatGPT. How would I word it? It remains ambiguous whether the upregulation of transcripts encoding mitochondrial proteins results in a net increase in like signals a net increase. That's just kind of a weird turn of phrase. Again, like weirdly colloquial, which is not the first time I've mentioned that in this paper. Oh my god, I'm seeing the argument for it being ChatGPT, right? Like, why not just cut the fat out of that sentence? Whether the upregulation of transcripts encoding mitochondrial proteins results in a net increase in mitochondrial mass. What's with signals a net increase? Like what does that mean? Signals a net increase? Like that, I know this is pedantic, but this is a weird way to phrase this, like a really weird way to phrase this. Um, and unless you've been reading papers for years, I don't think it would register as weird, but signals a net increase is unnecessarily it obfuscates the actual relationship. Does it make thing bigger or not? That's all you needed to say. Signals a net increase is not even the first way you would think to describe this, right? Like you had to that's you had to try for that one. That's so weird. Again, I'm only picking on the language because other language in this paper has annoyed me as well. Okay, so is it making mitochondria bigger or is it healing the mitochondria somehow? It's like they were given a hard word count limit from nature and had to cram a 30-page paper in only 10 pages. Every sentence is just trying to do so much lift. I actually disagree, Clockwork. I think a lot of these sentences are very fluffy. Like signals in that increase is fluff. Given this wealth of evidence is fluff. It is not surprising is fluff. A lot of these are extra that you could cut out and have no loss in efficacy of communication. Okay, so in order to determine if mitochondria got bigger, we imaged an independent series of experiments, imaged the neurons. This is also weird. Like, I just kind of assume that, yes, you're not imaging the same neurons using two different microscopy methods. That's just kind of a given, but whatever. Confocal laser scanning microscopy is a technique where you take slices of an image, so you can zoom the focus. Sorry, not the focus. You can zoom in. You can choose your field of your plane of view. Essentially, you can imagine confocal microscopy as you are only collecting light from a certain slice of an image. I love confocal microscopy. You can go up and you can go down. You can take slices. You can reconstruct those slices into a 3D image. It's very, very cool. I don't know what optical photon reassignment microscopy is, so I'm just gonna have to take their word for it there. Yeah. Both forms of light microscopy were validated against ultrastructure colon? What? Colon? Were validated against ultrastructure colon. Automated morphometry of deconvoluted image stacks established that although both optical methods overestimated the true organelle size to some extent, the size ratio of mitochondria in these neurons and uniglobular olfactory projection in neurons of the antennal lobes matched that determined by volume electron microscopy. One more time. Uno más. One more, please. So this is saying that we looked at our light microscopy and then we looked at some other method of looking at the neuron to see that they were about right. Sure. Although diffraction, why the colon there? What is going on? Ultrastructure, automated morphometry. Like what? It's a nature paper, man. Why is there a colon there? Sorry, I'm gonna move on. Although diffraction undoubtedly inflated the absolute dimension of mitochondria, especially in these images, valid inferences about relative morphological differences could therefore still be drawn. Although diffraction undoubtedly inflated the absolute dimension of mitochondria, especially in these images. Oh, no. Wait, chat. Check my sanity here. This sentence just doesn't make any sense, right? Although indicating that there is about to be a there's about to be a pivot. Although diffraction undoubtedly inflated the absolute dimension of mitochondria, especially in these images, Valid inferences about relative morphological differences could therefore although diffraction undoubtedly, there should be extra words here. You accidentally a word. You accidentally a whole bunch of words. Okay, this should be, although diffraction undoubtedly inflated the absolute dimension of the mitochondria, especially in these images, valid inferences about relative morphological differences could therefore be drawn due to something up here. Because this sentence, this isn't about the science. This is purely about writing sentences. The construction of this sentence is missing the justification, which I assume is up here. But if you wanted to read not like a crazy person, you need to put the reason that you have the valid inference here. Like why is this written this way? This is breaking my brain. This is one of the sentences ever written. Yeah. My images are blurry, but I can still see the size differences, is what that says, I think. Yeah, that is what that is saying. But in terms of like, you are led to believe that they're about to explain why valid inferences can still be drawn. It's not anything to do with the science. It's just the order of words, the order of sentences could be improved here. Nature seems to recommend 4,500 words, which are short. Yeah, Nature papers don't like fluff. Like, you're allowed to be more narrative in Nature paper because Nature papers are built around style, like having a very narrative style. But a lot of this is unnecessary for the style. I'm just picking on their writing. I don't want to say that they're bad scientists. Just the writing is confusing. Although diffraction undoubtedly. You know what it is? It's because there's could therefore still be drawn. Therefore implies that you have given a reason in this sentence. Because it starts with although. It's just a very confusing sentence. It's just a very confusing sentence. Then why not say blurry or limited resolution? Why bring diffraction into it? It's for microscopy nerds to criticize because you want to be as technical as you can when describing results. This is a case where, like, I think the jargon is fine. I just think that this sentence is particularly clumsy. You know, I'm calm now. I don't love the sentence. It's just compounding, right? Every time I stumble on a bit of communication, I get a little more irritated. Because this is also trying to cover their own butts, right? This is saying, okay, our results are like not perfect, but like it's still pretty chill here. So trying to maybe obfuscate or excuse some imperfections in their data collection, which is understandable to a certain extent. But again, combined with the other language used in this paper, like, you were way too cocky given this wealth of evidence, and then you oh, our pictures were perfect. Like, just don't have that tone in there and it's not a problem. A night of sleep loss, regardless of whether a night of sleep loss, regardless of whether caused a night of sleep loss, regardless of whether caused by mechanical agitation. Speaking of stumbling over a sentence, a night of sleep loss, regardless of whether caused by mechanical agitation or an artificial elevation of arousing dopamine, reduced the size, elongation, and branching of the sleep neuron mitochondria, and led to the relocation of dynamin-related protein, the key fission dynamin of the outer membrane, from the cytosol to the mitochondrial surface. Okay, we'll get into that when we look at the figure. The PRM, which achieves lateral super resolution approximately twofold above the diffraction limit, detected a concomitant increase in mitochondrial number indicative of organelle division, which eluded the confocal microscopy. Extended data, but see methods for a cautionary remark about the interpretation of absolute mitochondrial count. What? Huh? Are you talking directly to me? I've never seen language this casual in a nature paper before. Did we all agree to write papers more casually at some seminar or something or some big thing? This is crazy. See methods for a cautionary remark about the interpretation of absolute mitochondrial counts. Ugh. My eyebrows are going crazy. I actually don't love casual writing. I know I disagree with a lot of people that think that scientific papers should be more accessible to the public or to non-experts. Um, I just think there's a better way to do this, right? If the argument here is see methods for cautionary remark about the interpretation of absolute mitochondrial counts, instead what you could do here is mitochondria, uh, methods of mitochondrial counting. I'm paraphrasing here, but methods of mitochondrial counting have some pros and cons, and then put a citation where some other person has a methods paper about interpreting mitochondrial counts, because this is like, trust me, bro, right? See methods, so see what we did for a cautionary remark about the interpretation of absolute mitochondrial counts. Surely, if this were an issue, someone else would have done it, and then you would have been able to cite it here. It's very, very bizarre. This seemingly incomplete tally of daughter mitochondria notwithstanding, the two methods painted congruous pictures of mitochondrial fission after sleep loss. The mitochondria of antennal lobe projection neurons, by contrast, bore no vestiges of sleep history. Bore no vestiges of sleep history. Again, just very, very flashy. Just say number go up or number go down. Like, this is a very confusing paper to be read. It's like jarring. You know, the language choice is jarring. Bore no vestiges of sleep history. Bro, we're not affected by sleep. Bro. Yeah, this is something that you would put in a press release or something, not in a paper. AOX protected the sleep neuron mitochondria against sleep loss induced fragmentation, underlining that the ROS generation during waking is the initial spark that triggers fission. In the same vein, depolarization of these neurons during mechanical sleep deprivation, which increases ATP consumption by the pump and thereby reduces the diversion of electrons into the ROS, preserve the morphology of their mitochondria. Mitochondrial fission is a prelude to the proliferation of mitochondria at contact sites with the endoplasmic reticulum and or shedding and clearance of dysfunctional organelle fragments by mitophagy. Sleep deprivation stimulated both processes. The reconstitutional fluorescence from GFP fragments anchored in the outer mitochondrial and endoplasmic reticular membrane, whose mere presence had no effect on sleep, showed higher contact site counts in these neurons of sleep-deprived flies, whereas MitoQC, a ratiometric sensor detecting detecting the entry of mitochondria into acidic autophagal lysosome, reported enhanced mitophagy. Mitochondrial endoplasmic reticulum concentrates the fission and fusion machineries whose dynamic equilibrium determines the steady state morphology of the mitochondrial network and support mitochondrial biogenesis by allowing the passage of phospholipids from the endoplasmic reticulum. Okay, this is a lot to take in. So, mitochondrial fragmentation, mitochondrial fission is just like mitochondrial reproduction. It's like when mitochondria split. I don't actually know what the biological context of mitochondrial fission is. Maybe it's a detection of like increased energy demand, but I'm not entirely sure. Yeah, fission is a prelude to the proliferation of mitochondria. So it happens before you get reproduction of the mitochondria. Oh, so what is this supposed to mean? A night of sleep loss stopped mitochondria from growing, and it's key fission from the outer membrane from the cytosol to the mitochondrial surface. Organelle division, which eluded the confocal microscopy. I don't know why it would have been in one form of microscopy, not the other, but whatever. I'm not a microscope expert. The two methods painted congruous pictures of mitochondrial fission after sleep loss. So apparently, when you are very eepy, you have more mitochondria. Okay. AOX. Don't remember what this is. What is AOX? Alternate oxidase. I see. So this is when you are helping reduce the stress on the electron transport chain machinery. That protected these mitochondria against fragmentation. So they no longer do the fission thing. So they no longer reproduce, split. Underlining that the electron transport, the reactive oxygen species generation during waking is the initial spark that triggers fission. I don't know about initial spark that triggers fission. Why not just say that it is involved in triggering fission? Because this is saying if you protect the electron transport chain by giving them a release valve, this protects against mitochondria splitting. Underlying that reactive oxygen species generation during waking is the initial spark that triggers fission. You could say instead that this result is consistent with previous research that shows the same thing. It's, again, kind of weirdly worded in my opinion. In the same vein, depolarization, so when you fire neurons, when you fire these sleep neurons during mechanical sleep deprivation, which increases ATP consumption by this pump, and that preserve the morphology of the mitochondria. So this is getting really hairy, but when you force the sleep neurons to fire, that makes them sleepy and also prevents mitochondria from splitting. Mitochondrial fission is a prelude to the proliferation of mitochondria at contact sites with the endoplasmic reticulum. Okay, that's fine. Okay, data. If sleep loss makes more mitochondria, can you lift more weights or otherwise generate more power with sleep deprivation? This is an excellent question, Leo. Unfortunately, this is only looking at the subset of neurons within the brain that control sleep in the fly. So I've been pretty good throughout this paper reminding the audience that we are specifically looking at just the Drosophila sleep neurons and not anything more than that. Yeah, I know. Depriving yourself of sleep is not the way to get your gains, unfortunately. Volumetric renderings and morphological parameters of automatically detected mitochondria in the fancy image thing of these dendrites and rested flies. Sleep deprived flies recover 24 hours after sleep deprivation. Volumetric renderings of the mitochondria. Okay, so we're using super fancy technology to see what the shape of the mitochondria are in these neurons. So on the left here, we have rested. On the right, we have sleep deprived. And on the right, we have recovering from sleep deprivation. So what do we think, chat? Let's compare these two, rested and sleep deprived. We're looking at just the mitochondria. And so what do your eyes tell you about the mitochondria in the sleep deprived neurons? What do they look like? Or how would you describe the difference here? Left versus right, sleep deprived and well rested. Dots. They are all certainly dots, but do your eyes tell you something different about rested mitochondria versus sleep deprived mitochondria? Anything stick out at you? And again, you don't have to have any, you don't have to have a background in biology or any fancy language. Just do your best. The difference is that there are fewer dots in the sleep deprived one or the rested ones. And again, if you are looking to get into biology, you should be comfortable with describing what you see in the data. Because I know what I would describe left versus right. maybe I'll zoom in a little more. Maybe that'll help you. Um, I'll make this a little more full screened. How is that? Right, so comparing left and right. I mean, what are those dots supposed to represent? So each dot is a mitochondria. Yeah, just the mitochondria. We are just looking at the shapes of the mitochondria. So what can we say about the difference between the shapes of the rested mitochondria and the shapes of the sleep-deprived mitochondria? What can we say here? Okay, if we zoom in here, maybe this will be a little easier to digest, right? I'm focusing on the rested ones. You can kind of see what the shape of these mitochondria are. And then I will pan over the sleep-deprived one. Sleep-deprived has fewer / smaller mitochondria. They look bigger and overall, they look bigger and more in the rested one. Yeah, I would agree. Like, just by eye, I would say that there are more mitochondria in the rested state and fewer in the sleep deprived state. And then in the recovered state, you can see these mitochondria, they got big. They got pretty big. Like, recovered from sleep deprivation, they got big. I'm beginning to see why they needed to hedge their results so much, because from our eyes looking at this, right? I think there are more here, but I think they're going to argue that there are actually more here and that you can't rely just on microscopy to count mitochondria. I think that's why they were hedging. So we'll see that in the sleep deprived. Oh no. Yeah. That's what they're saying. To me, it looks like there are more mitochondria here, but when you count them, apparently, in the sleep deprived ones, this purple group, you have more mitochondria. Sure, machines can count better than human eyes can't, but to me, it really does look like there are more mitochondria in the rested. And the volume, we certainly can see that in the sleep deprived here in pink. Sorry, let me scroll down. Yeah, so number of mitochondria, the pink represents the sleep deprived guys. So yeah, there are more mitochondria in the sleep deprived, which is not what our eyes tell us, but yeah, maybe that's just like a machine thing. We do see, and I think this is accurate from what we're seeing with our plain eyes, is that sleep deprived mitochondria are smaller. So they are smaller and they are more numerous according to machine counting. And they are more spherical, right? So this pink right here, they are more sphere-like than mitochondria that were well rested instead of long. I don't know what branching is in mitochondria, but I'm going to kind of ignore that. So I think they're making the argument that because there are more mitochondria in the sleep deprived samples and that they are shorter, that is indicative of mitochondrial splitting, that the mitochondria are multiplying. Sure, I think that's fine. So sleep affects mitochondrial number. I think that's fine. Whether or not mitochondrial number affects sleep, that's, different. Different question. Yeah, what is C here? We have D here, but where is the caption for C? A, B. Hello? Where is C? Oh, here. DRP1 recruitment. Okay, so this is the protein that localizes to the middle of the mitochondria and splits it in half. So when you have sleep deprivation, the DRP protein fluorescence increases. Okay, so this is, I think, pretty decent evidence to show that when you are sleep deprived, you get more mitochondrial reproduction, essentially, because not only do you have more mitochondria, not only are they smaller, but you also have an increase in DRP1 fluorescence at the mitochondrial membrane. So this ring, this protein ring that cinches the mitochondria and cuts it in half, you get more of that when you're sleep deprived. Wait, sleep deprived has more but smaller mitochondria. Did they divide? That is what the paper is arguing. I think that's fair. I think that makes sense. And then D here, mitochondrial endoplasmic reticulum contacts, isosurface renderings in distal neuron branches obtained by trilinear interpolation of threshold and despeckled confocal image stacks. Okay, a lot of fancy words. Sleep deprivation increases the number of splix puncta per dendritic field. I don't know how this experiment works, so I'll just nod and say okay. So it looks like mitochondria are touching the endoplasmic reticulum more frequently when you are sleep deprived. Okay. In the sleep cells, of course. Okay. That's chill. I think that's cool. So it looks like mitochondria pop off when you are needing sleep. Okay, very cool. Into acidic autophagal lysosomes reported enhanced mitophagy. So where does that come from? Sleep deprivation, reconstitution from GFP fragments anchored in the outer mitochondrial endoplasmic reticulum membranes, whose mere presence has no effect of sleep. Higher contact sites in sleep deprived flies, whereas MitoQC, a radiometric sensor detecting the entry of mitochondria into acidic phagal lysosomes, reported enhanced mitophagy. So mitochondria are not only being made more, but they're also being eaten more, which is kind of interesting. Mitochondria endoplasmic reticulum contacts concentrate the fission and fusion machineries whose dynamic equilibrium determines the steady state morphology of the mitochondrial network and support mitochondrial biogenesis by allowing the passage of phospholipids from the endoplasmic reticulum. The abundance of mitochondria endoplasmic reticulum contacts in sleep deprived sleep neurons may thus not only echo a recent wave of mitochondrial fission, but also prefigure, weird word, as we suspect the abundance of transcripts from mitochondrial proteins does. As we suspect the mitochondrial protein does, the proliferation and fusion of mitochondria during subsequent recovery sleep, which cause their volume, shape, and branch length to rebound above baseline levels. Interesting. So this is saying that when you sleep, recovery, when you sleep, is it above? Okay, here it's above baseline levels. So you get sleep deprived, your mitochondria split and reproduce, and then when you sleep, when you recover, the mitochondria actually fuse back together. I don't really buy that because this data doesn't show that that's statistically significant. Like this sleep deprived mitochondria, which are numerous, they do look like they go down, but I don't see a statistical comparison between these two. So yeah, that's kind of weird. Volume, the mitochondria apparently remain large, or they get larger. Okay, I like that though. They get larger. Maybe they grew or maybe they fused. Without knowing if the numbers actually changed significantly, we can't really say. So they're still making conclusions that you should not be making, because their argument is that the mitochondria, when you are sleeping again after sleep deprivation, is a result of your mitochondria fusing together. But we're not seeing a statistically significant drop in the number of mitochondria when you are recovering. We see an increase in volume, which indicates that either the mitochondria could have grown or they could have fused. But without this piece, I would be pretty wary of making that conclusion. Yeah, I wouldn't be that comfortable with making that conclusion. Where was I? I got lost. Oh no. Okay, I'm here. Okay. So this, in terms of the overall argument, do mitochondria drive sleep? This is more like sleep affects mitochondria. So I'm not really too convinced about anything about that in particular so far. But here's the reverse of that, right? So we know that sleep, so this is saying sleep affects mitochondria, and now this is saying mitochondria affects sleep. So this is what I'm more interested in. So how are you going to do that? If shifts in the balance between mitochondrial fission and fusion are part of a feedback mechanism that corrects mismatches between this high energy electron carrier supply and ATP demand that causes sleep pressure to rise or fall, then the experimental induction of these homeostatic responses in these neurons should move the set points for sleep. Mitochondrial fragmentation is predicted to decrease, and mitochondrial fusion is predicted to increase sleep duration and length. To test these predictions, we took experimental control of these GTPases with central regulatory roles in mitochondrial dynamics. The fission dynamin DRP1 and integral proteins of the inner and outer mitochondrial membranes termed optic atrophy and mitofusion or mitochondrial assembly and regulatory factor, respectively, whose oligomerization and cis and trans fuses the corresponding membranes. Oh boy. Okay. So, what does this mean? This means they are predicting that if you force mitochondria to fuse, then you are going to get sleepier. And then if you force the mitochondria to split, you are going to be sorry, this is confusing. Mitochondrial fragmentation is predicted to decrease, fusion is predicted to increase. So if you fuse them together, you are going to sleep more. And if you break them apart, you're going to sleep less. So if we want to answer the question, do mitochondria drive sleep? It might not be a bad line of evidence to show if you remove the mitochondria from a cell, then you no longer sleep. I'm beginning to accept that as being a line of inquiry. Mitochondria split and fuse, they do. They do, in fact, do those things. Although I'm not an expert in understanding how that particular mechanism actually works. Let me shrink this PDF real quick. Maybe just a bit more. Okay. Okay, so this is one way that we can directly test the relationship between mitochondria and sleep, because if you selectively do something to the mitochondria and that affects your sleep, I'm much more open to this being a feature of mitochondria versus just a feature of having energy around, right? Especially if the ATP levels don't change over time. But how are they going to prove that? Fragmenting these sleep neuron mitochondria through the overexpression of the mitochondrial DRP1. Okay, so this is the mitochondrial cutting protein. This is a protein that forms a ring, I believe. Someone correct me if I'm wrong, but I think this forms a ring inside the mitochondria, and then that ring squeezes. And you can imagine like a tube of toothpaste that squeezes the mitochondria in two. Or the RNAi-mediated depletion of OPA1, and to a lesser extent of MARF, decreased sleep. So I don't know what OPA1 or MARF are, but I'm probably mostly going to pay attention to DRP1. Abolish the homeostatic response to sleep deprivation and reduce ATP concentrations regardless of sleep history. Tipping the equilibrium towards mitochondrial fusion had the opposite effect. The restricted knockdown, or DRP1, or the overexpression of OPA plus MARF, or OPA alone but not MARF alone, increased baseline as well as rebound sleep, and elevated the arousal threshold without causing overexpression artifacts or overt developmental defects. I guess I'll be the judge of that. None of these interventions altered sleep when targeted to projection neurons or Kenyan cells. Okay, very cool. I do like that. The indiscriminate sleep losses reported after pan-neuronal or glial RNAi knockdowns, either DRP1 or MARF, which in DFBNs alter sleep bidirectionally, reflecting their antagonistic mitochondrial shaping roles, are difficult to interpret without parallel measurements of mitochondrial form and function. Taken at face value, they could hint that the fusion-fission cycle of many non-sleep cells are not tied to sleep, but sleep-wake cycle, but operate continuously to mix and recompartmentalize mitochondrial content for maintenance or metabolic control. Boy, okay. So this is, it's tricky because I'm kind of falling for the argument here, because it's kind of funky, right? So this is saying that if we just change the number of mitochondria by promoting fusion or fission, then that affects sleep. And to me, that's wacky, right? That's not a direct ATP changing thing, although ATP could be changed, but just having the number of mitochondria change whether or not you get sleepy is kind of crazy. So this is cyclical, right? There's a cycle. When you get sleepy, you get more mitochondria, and then when you sleep, you get less or fewer mitochondria, supposedly, right? We saw that they got bigger. We don't know if they actually fused. Statistically speaking, we have to be very careful about, again, like drinking their Kool-Aid. But to me, this synchronicity of like, okay, when you are awake, your mitochondria are big and healthy and strong, and then when you are feeling sleepy or sleep deprived, your mitochondria split. To me, this is a much closer relationship between the existence of the mitochondria, like this weird fusion fission cycle, if that is coupled with sleep, independent of ATP production, I think that would be pretty cool. I think that would be pretty cool. But we're just gonna have to see what that is like. Oh, okay. Let's continue. In these sleep neurons, the large and opposite behavioral consequence of promoting mitochondrial fission or fusion went hand in hand with established biophysical signatures of lower high sleep pressure. These sleep neurons, in short sleepers, overexpressing the mitochondrial cutting protein, had shallower current spike frequency functions than neuronic control animals, whereas the converse was true in somnolent overexpressors of OPA1 and MARF, whose DFBNs generated an elevated number of somnogenic bursts as a part of their enhanced responses. So this is saying that when you are cutting up the mitochondria, you have a certain change in spike frequency functions in neurons. I don't know if that is shallower. So this is saying that if you start cutting up the mitochondria, you start forcing them to divide, then you stop being able to sleep quite as well. Whereas if you overexpress the fusion proteins, then you have better sleep activity. So this is not in the actual full fly, but this is just the action of the sleep neuron. So we got to make sure that we actually see an animal come out of this as well somehow. Okay, so now we can look at the figure here. Yeah, so okay, they have a figure here. I was right. DRP1 is a protein that localizes to the center of mitochondria, and they form this kind of ring, and that ring contracts. And when that contraction finishes, you get a cutting of the mitochondria. Whereas MARF and OPA apparently are proteins that force the mitochondria to fuse. So what do we got here? A whole bunch of stuff. So this is measuring sleep in the light cycle and the night cycle, I'm guessing. What is B here? Sleep profiles in flies with fission or fusion proteins. So in the teal, we have the knockdown. So this is going to be more the knockdown of the fusion means that there will be more fission. So we are splitting up the mitochondria more frequently, and as a result, they don't sleep quite as well. That is consistent with the sleep-deprived neurons having more fizzed mitochondria. And the reverse is true when it is overexpressed. So the more you force your mitochondria to smash into each other, the increased need for sleep happens, right? In the light and in the darkness, you get more sleep. That's what this blue is. And I don't really know what these gray lines in the middle are, to be honest. I believe that we'll see that that is true up here as well. So the promoter is good. This is overexpression of the fission protein. That is lower than just... Oh, don't quite understand that. My brain is fizzing out, man. I've been streaming for how long? Oh my gosh, almost 4 hours. Oh god, we gotta finish this soon. Okay. Oh, it's Zeitgerber time again. Fascinating. Yeah. Let's see. These are the spike potentials, but they don't finish with actual... Okay, so they feel... Okay, so these are the actual sleep for 30 minutes. Okay, so these are actual flies. Okay, so this is showing that as you fuse the mitochondria together, as evidenced by the blue line, you have an increased need for sleep. And when you when you cut them, they have a decreased need for sleep. That's pretty interesting. These are in actual animals, so that's cool. That is pretty cool. I want to believe this paper so badly is the thing, so I think it's really cool. There are certain ways that this paper was written that really bother me. In flies expressing this driven fusion of protein, regulating parental controls, manipulation that increase fission or fusion after sleep in opposite directions. DRP1 RNAi. Also, I hate how this figure is made. I don't think it should be dots. It's kind of hard to see where these line up. I think they should have like lines to help actually tell where ones begin and end. Yeah. Yeah. Tough. Pretty hard to figure out here. Okay, so percent of sleep lost. So sleep deprivation. This what is D? What are we looking at here? D. Manipulations that increase fission or fusion alter the time courses of sleep rebound after deprivation in opposite directions. Sleep regained. So if you cut out the fission protein, which means you have more, if you cut out the fusion protein, meaning you have more fission, you get a very small, these are not big changes. But I think what's most important is that when you overexpress the fusion proteins, you get a lot of sleep return, so percent sleep regained. Okay, so this is all consistent with this idea that fusing mitochondria together drives sleep, and then splitting them is associated with sleep deprivation. Hang on, I don't quite follow. No sleep equals split mitochondria. Yes sleep equals plump mitochondria. DRP force splits mitochondria equals bad sleep. OPA1 force fuse good sleep. But shouldn't DRP1 make you feel tired and induce sleep? Yes. Hold on. Yeah, it's difficult because it's like it's correlation and causation, or chicken or egg kind of situation. I think it is that when you don't sleep, you get split mitochondria, indicating that split mitochondria are begging you to sleep, which is what Leo, you said. So here, if you see DRP1 RNAi, we'll take this one. If you force them to make extra, this. Oh, this is so confusing. Hold on, you make an excellent point. I need to slowly read that back again. Indiscriminate sleep loss is reported after pan-neuronal or RNAi glial knockdown bidirectionally, reflecting their antagonistic. Well, that doesn't help, but okay, here. Fragmenting the mitochondria through the overexpression of DRP1 decreased sleep. So this is causative. So you are so what is driving the lack of sleep? I could use some sleep right now. I slept wonderfully. It's just 4 hours of wrestling with this paper. I feel like I'm in graduate school again. So we just took some mice and then we forced them to not sleep. You see that the fission is active because the fission is what happens when you are sleep deprived. Now, why does it happen when you are sleep deprived? Is it a cause or an effect? Let's take a look here. When you manually fragment the mitochondria, you did not sleep as much. Abolish the homeostatic response to sleep deprivation. 4B. That's this. And that's what I said. That's what I said, right? You knock out the fusion one and you sleep bad, which is the same thing as overexpressing the fission one. Okay, I need to write this out in chat. So when sleep deprived, fission is up. When you force fission to happen, you do not sleep as much. So there is something about not sleeping and the mitochondria splitting that is true. And then the converse is also true. When you fuse them together, you sleep more. Yeah. Leo, does that make sense? Or have I just made things more confusing? Because I'm trying to say out loud as well. It's not so much tiredness. I think tiredness is maybe something different. This is purely about uh, the actual amount of sleep gotten, which I think is slightly different than the physiological perception of tiredness. Because I agree with you, that would make sense, like sleep induction. But you also have to keep in mind that, yeah, I think so. Yeah, yeah, because I don't think we can think of it as sleepiness. I think we have to think of it as driving sleep, which is just different enough to be super confusing. But I think that's what this paper is trying to say. Mitochondria splitting is associated is causing you to not sleep, and then mitochondria fusing is causing more sleep. Yeah. A striking feature of sleep deprived brains is the depletion of phosphatidic acid, a fusogenic glycerol phospholipid. Mitochondrial phosphatidic acid is a cleavage product of cardiolipan. I think somebody in chat had mentioned this, generated by a local phospholipase D, underlining the importance of phosphatidic acid for fusion reaction and of mitochondrial fusion for the regulation of sleep. These interference, blah blah blah. Ooh, zucchini. Which stabilizes catalytically active mito-PLD or other transfer phospholipids from other cellular membranes to mitochondria, recapitulated the sleep loss seen when the protein-based fusion machinery of these neurons were targeted by RNAi or antagonized by the overexpression of DRP1. Okay, I think it's all saying the same thing. Right? Yeah. Fission stops sleep. Fusion happens when sleep. So fusion is what is driving the desire to sleep, or driving the physiological desire to sleep in these neurons. Do I have that correct? Let's read the discussion. So this is, oh my god, this is a very short discussion section, which is actually kind of nice. So let's see if our conclusions are consistent with what the authors are saying to see if we interpreted this information correctly. Aerobic metabolism was the innovation that, following the first two large increases in atmospheric O2 levels, 2.4 billion and 7,500 to 570 million years ago, allowed eukaryotes to maximize the free energy yield of electron transfers, setting the stage for the Cambrian explosion of multicellular life in the wake of the second O2 revolution. Power-hungry nervous systems appeared, and with them, apparently, the need for sleep. Although sleep is likely to have since acquired additional functions such as synaptic homeostasis or memory consolidation, an empirical power law that relates daily sleep amount to mass-specific O2 consumption suggests that sleep serves an ancient metabolic purpose also in mammals. I wouldn't call mammals ancient, man. When I think ancient, I'm thinking like archaea, or like cyanobacteria, right? Mammals are not that old. The allometric exponent in this power law is a multiple of 1/4 rather than 1/3 expected from Euclidean geometric scaling. A sign that the this is somebody got too math happy here. A sign that the distribution of resources by centralized networks such as the vascular and respiratory systems is responsible. Okay. Power-hungry nervous systems appeared, and with them, apparently the need for sleep. I agree. Although sleep is likely to have since acquired additional functions, an empirical power law suggests that sleep serves an ancient metabolic purpose also in mammals? Hmm. Look, I'm not like a math guy, right? So I don't really know how legit this is, but this is like an eyebrow raise for me just because I don't have this background, right? This is proposing that sleep it is making the assumption that um sleep scales to empirical power law. You sleep, okay, so this is saying sleep and energy consumption are linked because the more O2 you consume, the more sleep you need, which is not controversial. I think we all had this general idea that sleep is good for getting your energy levels back, which is kind of like, I don't know, obvious is not the right answer, right word, but it's not surprising, I guess, that sleep and energy. But the thing is the thing I take umbrage with is, you know, why take this swing to begin with, right? Why not just say energy consumption is related to sleep, which is what I think a lot of this is, apart from the mitochondria fusion thing, which is really cool. But yeah, bizarre, whatever, bizarre. Like if you want to say mitochondria equals O2 consumption equals sleep, sure, but I think that's pretty narrow-minded and kind of clickbaity, but whatever. Um, the allometric exponent in the power law is a multiple of 1/4 rather than 1/3 expected from Euclidean geometric scaling, a sign that the distribution of resources by centralized networks such as the vascular and respiratory system is responsible. I don't know what any of that means. This is some math person stuff. So I'm going to take my biology only person dunce cap and I'll just wear that and say sure. There might be some mathematical relationship. Okay, but I don't get it though. Wait, why? Why would this point to? Okay, whatever. If anybody has insight into this, I would love to know because I don't know what connection the authors are trying to make here. I might be just genuinely be too stupid. Thanks to higher terminal branch densities, these networks allocate more O2 to each cell in small animals, allowing their metabolisms to run hotter than that of large mammals whose cells are supply limited. Okay, I have heard of this. That is something that I have heard. The price to pay is a shorter life, a greater fraction of which is spent asleep. Okay. Are there smaller animals that outlive humans that have um a different relationship to sleep? This seems kind of like a generalization I'm not prepared to back up. Even within species, variations in sleep requirements of individuals, including those with identical nuclear genomes, such as our own isogenic flies, can arise in part from differences in the resistance to electron flow or of respiratory complexes containing subunits encoded by mitochondrial DNA. An overwhelming sense of tiredness, which is unrelated to muscle fatigue, is in fact a common symptom of human mitochondrial disease. Sure. I mean, you're allowed to be speculative in the discussion. It doesn't really make me mad. I just don't necessarily buy it. Yeah. The naked mole rat lives quite long. Then I'm wondering what if their story is real, then the naked mole rat must have some pretty unique mitochondria. Maybe a future stream. If sleep indeed evolved to fulfill a metabolic need, it is not surprising that neurons controlling sleep and energy balance would be regulated by similar mechanisms. In the mammalian hypothalamus, the mitochondria of orexigenic neurons expressing agouti-related protein and of anorexic genic neurons expressing pro-optomelanocortin undergo antiphasic cycles of fission and fusion. These cycles are coupled to changes in the energy balance of mice. Just as cycles of mitochondrial fission and fusion in these sleep neurons are coupled to changes in the sleep balance of flies. Hypothesis: if you pump an organism full of OPA1 to counteract the sleep deprivation effect in caffeine, you can stay awake indefinitely. Maybe. Because this is making the argument that it's leaking from electrons leaking from too much ATP when you are uh, 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 freshly awake. That's causing some chaos, or sleep deprived that's causing some chaos. Um, I think the way to look at it is that there are special neurons in your brain that are itching to fire to make you sleep, and if you aren't sleeping, those neurons accumulate ATP, and then the electrons overflow, and then it's that electron flow that actually drives sleep somehow. So if you and then that's like they don't really connect that to the mitochondrial fusion and fission. It's like two separate levels of evidence, but they don't really talk to each other. So it's kind of hard to put it together in my brain. The electrical output of these neurons increases after mitochondrial fusion to promote weight gain and fat deposition. Huh? The electrical output of these neurons increases after mitochondrial fusion to promote weight gain and fat deposition. I'm guessing this is literal weight gain and fat deposition, like the mice are getting heavier or something, or flies or whatever. Just as the electrical output of these neurons increases after mitochondrial fusion to promote sleep. Deletions of mitofusins from these neurons impair the consumption of food, just as interference with mitochondrial fusion impairs induction of sleep. These parallels suggest that sleep pressure and hunger both have mitochondrial origins, and the electron flow through the respiratory chain of the respective feedback controls like sand in the hourglass that determines when balance must be restored. I mean, that's a pretty sick way to describe that. I like in the discussion you are allowed to be fluffy. So this analogy is great. Like feedback and cycles like sand in the hourglass that determines when balance must be restored. I think that is fine. I don't know, Clockwork, if you were expecting me to get mad at this discussion section, but to me, discussions are where you can wax poetic and take wide swings. And, you know, even if I don't buy what they're saying, like, I don't really mind that they are saying some crazy stuff. Now I feel like dropping this paper in the Discord was an act of violence. Again, I don't think this discussion is that bad. Like, it is like the discussion section, it's okay to like make comparisons and to wax poetic. So this is essentially saying that like the mitochondrial cycle of fission and fusion controls appetite and metabolism, and this fission fusion cycle also drives sleep in these neurons, which I think is a fine parallel to point out, as long as you aren't like saying this must be the way that things evolved, which is what they did in the abstract, right? Like, this is fine to say here. To hint at this in the abstract, saying this is the only way things could have evolved from the mitochondria, I think that's too far. I think that's too crazy. Yeah. So let me work on a little PowerPoint for you all to summarize this very long journey. I don't know how many of you have been with me since the beginning of this stream, but let's maybe tab over to, I'm doing this at the last second. So let's go ahead and hide the PDF, and I will do a little summary of what I believe I understood. Although I don't know that I fully understood things. So I'm going to work on this PowerPoint for a sec. And in the meantime, I kind of want to hear from chat, like, what did you think of this paper? Like, you know, apart from the language, what did you think of the research? What was your experience of this stream? What was it like? Like, did you enjoy me, you know, in going into this paper? Was this paper an appropriate level of difficult? Was it too hard? Was it too easy? Et cetera, et cetera. So, yeah, I'd like to hear what you have to say. The hunger argument is a bit weird to me. Surely we'd also feel hungry if we hadn't evolved with the mitochondria. Yeah, that's why I think this is one of those like Newton laser space sword things where like there are some questions that are not worth asking because you can't reasonably test for them, right? Yeah. So let's see. So one, your so for flies, I'm typing this on a PowerPoint, which I'll share in just a second. For flies, sleep deprivation is associated with okay, so actually maybe even go back. The sleep neurons in the fire and cause you to sleep. Not sleeping, deprivation. Do you like this keyboard, by the way? It's very, very clicky clacky. I realize it might be kind of loud. Not sleeping, sleep deprivation, causes an energy build up in your sleep neurons. This energy build up is associated with leaky electrons because the supply of electrons is too high for the demand for ATP. I don't think that the authors proved that it's the electrons that the leaky electrons that are driving sleep. And then separately, mitochondrial fission, splitting / reproduction, is associated with not sleeping. Mitochondrial fission is associated with sleeping. If you force mitochondria to split, you induce, you prevent sleep. If you force mitochondria to fuse, you force sleep. Okay, I think that's it. I'm going to now cast this. There we go. That's better. Let me get the chat back up here. Okay, so this is the state of things, I think. This is the state of things. Sleep neurons in the fire, they cause you to sleep. If you don't sleep, that causes an energy buildup because those sleep neurons are just kind of hanging out and not firing. This energy buildup is associated with leaky electrons because the supply of electrons is too high for the demands of ATP. I don't think the authors ever proved that it was the leaky electrons that are driving sleep. They tried to get at it, but it's not as good as I would like that experiment to be. And then a separate line of inquiry had found that mitochondrial fission is associated with not sleeping. Mitochondrial fission is associated with sleeping. And these fission fusion cycles happen in synchronicity with your ability to sleep. So if you force the mitochondria to split, you prevent sleep. If you force the mitochondria to fuse, you activate sleep. You can think of it that way, I think. So if we go back to the title of the paper and ask, do mitochondria drive sleep? I'm not entirely, like, I think it's better. The evidence is more compelling than I gave it credit for at the beginning, but I still think it's crazy to say that this is the only way it could have happened with mitochondria, right? Like, there are just so many other things in there are a lot of animals, right? To just be like, to not bring up a single animal that might be the exception to the rule here is kind of like showing your whole rear end, right? Like, it's kind of sus in a way. So what do we think of this, y'all? What do we think of this? What do we think of this particular explanation for this particular explanation for how mitochondria affect sleep? Do you buy the author's argument that mitochondria are the reason why we sleep, or what do you take away from this? Let's do a bit of a check-in with the audience here. Yeah, what do we feel? What do you feel? How does this paper make you feel? If anybody would like to share. I could start by sharing what I feel, actually. I actually think that the science is very compelling. It's very cool that mitochondria have this clock, and speaking of clockwork, have this clock cycle where they fuse and split, and that is sufficient to drive sleep, right? I think where I take umbrage is in the insistence that this is an inescapable consequence of aerobic metabolism. That's just like not that's not giving nature and evolution enough credit for being incredibly creative. I'm sure that given a different set of initial circumstances, some organism out there in some universe, some galaxy, some planet out there, I'm sure there's some organism that can do aerobic metabolism that doesn't need to sleep, right? I don't like the interpretation, but the data I think are pretty cool. Gotta say. From what I gathered, it feels like the causation is inconclusive still, though the correlation is there. Yeah, I think the causation between mitochondria and the idea of sleeping, this is their like pet theory that they're pushing really, really hard, annoyingly hard. But like what like my advice is to say don't drink their Kool-Aid and don't use their language. Put it into your own words. My own words would be that that honestly the best result here is the mitochondrial fission and fusion thing, right? That this acts as a clock that is synchronized with your sleep. I think that's very cool and compelling. The stuff about the electron leakiness, I so want that to be something that they proved, but I think because they didn't shut off the regular electron transport chain, I just don't buy it. Maybe, as Clockwork said, giving them maximum credit, maybe it's a given that once you introduce that other proton pump that the electron transport chain just shuts down, but I don't know. There's no proof there, right? Because an additional pump may reduce the demand, like the flow of electrons through the electron transport chain, but they didn't test that either, right? Like it's a very simple step that they could have done that would have made me a little happier was to do something like you put in that artificial proton pump that reacts to photons, and then you test the flux of electrons through the electron transport chain somehow. I don't know how that would have to happen, but there has to be some metabolic measure that you can detect that can tell you about flux through that pathway. There just has to be. I refuse to believe that there's not a way you can do that. Because yeah, maybe introducing that pump, maybe the pump's not working so good, right? Like you know what? I take it back. What if it doesn't work? What if the proton pump that you installed just doesn't reduce the demand for the electrons through the chain? Did they test anything like that? I don't think so. Yeah, because the optogenetic data that we looked at was not the flux through the channel, but rather a channel rhodopsin to make neurons fire. So I don't know that they ever even tested how well their hydrogen pump from Archaea even worked in the flies. I'd be happy to show otherwise, but I didn't see it when I was reading through here. That's just mildly concerning. All right. Any questions for me? Any other comments that you have about this particular paper that I can help try to address? Yeah, it's just anything about the paper, or I'll be ending the stream soon because boy, 4.5 hours of streaming is a lot longer than I was expecting to be streaming for. So what do we think? What do we think? How did you react to the paper? I want to know. I want to know. I want to know what you all think about what work we've done today. I feel like you should rest. The mitochondria says so. That's fair. That's fair. It was a long time that we spent on this paper, but one that I felt was very fun. I did have a lot of fun, even though I did mauled earlier in the in this stream. With that being said, I will see you next time. I don't remember what I'm doing next week, but I am still working on these plaguing shorts. I have a very exciting script that I'm trying to finish this week and record and get done. I really want to say that I'll have it done by the end of August, but that may be kind of ambitious. But thank you all for coming. I hope you have a wonderful rest of your day, and I hope you allow your mitochondria to uh, use up the ATP in your sleep controlling neurons so that you do not have excess electron flux out of your electron transport chain, which could damage your neurons. And with that being said, goodbye, y'all. I don't think anyone else is streaming from VTheorem. Let me double check real quick so that I can raid people. I think we might be good. All right, bye-bye, bye-bye. Bye, y'all. Bye-bye.